the 193rd meeting of the Vaccines and Related Biological Products Advisory Committee. Please, for the committee members, be sure your microphones are muted except when speaking, and that your cameras are on while addressing the committee. This meeting, the committee will meet to discuss and make recommendations on the safety and effectiveness of Influenza vaccine mRNA manufactured by Moderna Incorporated, with a requested indication in biologics license application for the prevention of influenza disease caused by influenza viruses A and B represented in the vaccine. I will now read a statement regarding the Vaccine Advisory Committee Act and the Government in the Sunshine Act. For the advisory committee's committee topics, such as the one before us today, there may be a range of views indicating views individuals may hold strongly. Our goal is to ensure that the meeting provides a fair, orderly, and open forum for discussion, and views may be expressed without interruption. As a reminder, individuals may speak on the record when recognized by the chairperson. We appreciate everyone's cooperation in helping us conduct a productive and respectful meeting, consistent with the principles of the Federal Advisory Committee Act and the Government in the Sunshine Act. Committee members should ensure discussions related to the meeting topics occur in the open session and as part of the public record. Members are therefore reminded not to discuss the meeting topic during breaks, lunch, or other times, which would be difficult since we're virtual, outside the open meeting sessions. Thank you for your cooperation. I will hand the meeting over to Dr. Cecily Reese to provide administrative announcements, roll call, and reading the conflict of interest statements. Over to you, Cecily.
Thank you, Dr. Montel. Good morning, everyone. I am Cecily Reese, and I serve as the designated federal officer for today's meeting of the Vaccines and Related Biological Products Advisory Committee. Before we begin, I would like to thank the many individuals and teams who helped make today's meeting possible. On behalf of FDA, I extend sincere appreciation to the leadership of the Center for Biologics Evaluation and Research, as well as the leadership and staff of the Office of Vaccines Research and Review for their work in supporting today's meeting and the important scientific issues before the committee. I also want to thank the Advisory Committee Oversight and Management staff and our colleagues across the agency with whom we collaborate throughout the Advisory Committee process. These meetings require significant coordination, preparation, and support across many offices, and that work is essential to bringing forward a successful Advisory Committee meeting. We also extend a tremendous thank you to the AV team who make everything work for these meetings, and they also spend tireless hours putting meetings together. We also extend tremendous thanks to FDA's guest speakers for today's meeting. We appreciate the time, expertise, and information they will provide to support the committee's discussion. FDA also extends sincere appreciation to the committee members for their time, expertise, and independent judgment they bring to this important work. Finally, I would like to thank our acting chair, Dr. Arnold Montel, for leading today's meeting and for his continued service to the committee. To everyone who contributed to bring this meeting forward, thank you. I will next have the next slide, please. Please direct any press or media questions for today's meeting to HHS Press Room at hhs.gov/press-room. /index.html or 202-690-6343. The transcriptionists for today's meeting are Ms. Virginia Diaz and Ms. Mayra Angulo. Next slide, please. We will begin today's meeting by taking a formal roll call for the committee members and temporary voting members. When I call your name, please turn on your camera, unmute, and introduce yourself by stating your first and last name, organization, expertise, or role. And when finished, you may turn your camera off so we may proceed to the next person. Okay. First, we will start with Dr. Monto, who's chairing the meeting.
Good morning. I'm Arnold Monto. I am Emeritus Professor of Epidemiology and Emeritus Francis Professor of Public Health at the University of Michigan School of Public Health, where I have worked on the control and prevention of respiratory infections, principally influenza, for many years. Thank you.
Thank you, Dr. Monto. We'll next have Dr. Berger.
Hi, good morning. I'm Adam Berger. I am the Director of the Division of Clinical and Healthcare Research Policy at the National Institutes of Health. I'm a geneticist with additional training in immunology. Thank you.
Thank you, Dr. Berger. We'll have Dr. Durbin.
Hello, I'm Dr. Anna Durbin from the Johns Hopkins Bloomberg School of Public Health. I'm internal medicine and adult infectious diseases trained physician who conducts early phase and vaccine trials and challenge studies. Thank you.
Thank you, Dr. Durbin. We'll have Dr. Gans.
Hi, this is Dr. Haley Gans. I am a pediatric infectious disease physician at Stanford University, and I am the co-director of the Pediatric Infectious Disease Committee for Immunocompromised Hosts. My research focuses on the immune response to viral vaccines in several populations, including children, adults, and immunocompromised individuals.
Thank you so much, Dr. Gans. We'll now have Dr. Colmar.
Recording stopped.
Hello, my name is Dr. James Colmar.
Recording in progress.
I'll start over again. Hello, my name is James Colmar. I am an Associate Vice President, Therapeutic Area Head for Vaccines at Merck & Co. Thank you.
Thank you, Dr. Colmar. Dr. Munoz?
Yes, good morning. Flor Munoz. I'm a pediatric infectious diseases physician at Baylor College of Medicine in Houston, Texas. And I work as a clinician investigator in clinical trials of vaccines in special populations, including children, immunocompromised hosts, and pregnant women, and also work on vaccine safety.
Thank you so much, Dr. Munoz. We'll now have Dr. Omar.
Hi, I'm Saad Omar. I'm the founding dean of the Peter O'Donnell Jr. School of Public Health at UT Southwestern in Dallas. I'm an infectious disease epidemiologist, and my work has focused on epidemiological studies, clinical trials, and other studies related to vaccine preventable diseases and other infectious diseases.
Thank you, Dr. Omar. Dr. Asali?
Good morning. My name is Hannah Sahli. I am an infectious diseases physician at Baylor College of Medicine, Adult ID, and my clinical research focuses on clinical vaccine developments, phase 1 through 4. Thank you.
Thank you so much. Dr. Patel?
Good morning. I'm Manisha Patel. I'm the Chief Medical Officer in the National Center for Immunization and Respiratory Diseases at CDC. I'm a pediatric infectious diseases physician, and my area of expertise is in public health, specifically in vaccine preventable diseases. Thanks.
Thank you, Dr. Patel. And Dr. Perlman?
Good morning. I'm Stanley Perlman. I'm in the Department of Microbiology and Immunology at the University of Iowa and a specialist in pediatric infectious diseases. My long-term interest is in coronaviruses.
And thank you, Dr. Perlman. Dr. Rubin will not be joining us today. And I will now read the conflict of interest statement. Next slide, please. Thank you. The FDA is convening today's meeting of the Vaccines and Related Biological Products Advisory Committee, also known as VRBPAC, under the Federal Advisory Committee Act of 1972. The VRBPAC will meet in open session to discuss and make recommendations on the safety and effectiveness of influenza vaccine mRNA manufactured by Moderna Incorporated, with a requested indication in Biologics License Application STN 125.869/0 for the prevention of influenza disease caused by influenza virus subtypes A and type B represented in the vaccine. With the exception of the industry representative, the members of the committee are either special or regular government employees and are subject to federal conflict of interest laws and regulations. Accordingly, FDA's Office of Ethics and Integrity has reviewed the financial interests of the committee members for compliance with federal ethics and conflict of interest laws. The members were screened for potential financial conflicts of interest related to today's meeting agenda, both their own interests and those that are imputed to them, including those of their spouses, minor children, and employers. Based on the agenda for today's meeting and all financial interests reported by the committee members, the agency has determined that all members of this advisory committee are in compliance with federal ethics and conflicts of interest laws, and as a result, no conflict of interest waivers under 18 U.S.C. 208 have been issued in connection with this meeting. Dr. James Colmar of Merck & Company is participating in this meeting as a non-voting industry representative acting on behalf of regulated industry. Consistent with the current agency policy, FDA is only including industry representatives in advisory committee meetings where required by statute. FDA is required to include an industry representative in today's meeting under 22 U.S.C. 355N3C. Industry representatives are not appointed as special government employees, nor are they regular government employees. Industry representatives serve as non-voting members of the committee. Non-voting industry representatives represent all regulated industry and not any association, company, product, or ingredient, and bring general industry perspective to the committee. Under FDA regulations, although a non-voting member serves in a representative capacity, the non-voting members shall exercise restraint in performing such functions and may not engage in unseemly advocacy of or attempt to exert undue influence over the other committee members. There is a speaker from the Center for Disease Control and Prevention and a guest speaker at today's meeting who will give a presentation to the committee, answer questions from the committee, and hand the meeting back over to the chair. They will not participate in the committee deliberations, render advice to FDA, or vote. The CDC and guest speakers participating in this meeting are presenting the views of their organizations and not their personal views. The following guest speaker, Dr. Richard Webby, has declared no financial interest, and has been cleared to participate in the meeting. In the interest of transparency, FDA asks that guest speakers disclose personal financial involvement with a firm, product, or other entity affected by the committee's discussions to allow the audience and the committee to objectively evaluate their presentation. Today's guest speaker has not reported any such relevant interest. Consistent with the Federal Advisory Committee Act and the FDA Advisory Committee procedures, invited speakers who provide informational presentations and do not serve as committee members are not subject to the same financial conflict of interest screening requirements applicable to special government employees appointed to the committee. The speaker's role is limited to providing factual and contextual information to assist the committee's understanding of the subject matter. Accordingly, no conflict of interest determination under 18 U.S.C. 208 was conducted for these speakers. The speakers will not participate in committee deliberations or recommendations. FDA asks that all other participants, including the open public hearing speakers, advise the committee of any financial relationships that they have with any affected firms, its products, and, if known, its direct competitors. We would like to remind the members that if the discussions involve any products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participant needs to inform the DFO and exclude themselves from the discussion, and their exclusion will be noted for the record. Thank you, and I hand it back over to you, Dr. Montel.
Thank you. Let's begin by introducing Dr. David Kaslow, Director, Office of Vaccines Research and Review of CBER, who will give us our marching orders with an introduction. Dr. Kaslow.
Great. Good morning. Let me also extend a very warm welcome from the Office of Vaccines Research and Review to this 193rd convening of VRBPAC. So this short introduction will focus on level setting for today's charge to VRBPAC. Next slide, please. The agency asked VRBPAC to consider two applicant proposed approval pathways for a novel mRNA seasonal influenza vaccine. Both pathways require substantial evidence of effectiveness. Next slide, please. The first pathway is traditional approval. The applicant has proposed this pathway for an indication in adults 50 through 64 years of age. This is the standard approval pathway based on substantial evidence of effectiveness that generally comes from adequate and well-controlled clinical studies evaluating direct clinical benefit or a clinically validated surrogate endpoint. The evidence must establish that the benefits clearly outweigh the risks. The second pathway proposed by the applicant for adults 65 years of age and older is accelerated approval. This is an expedited approval that must treat a serious or life-threatening condition and must address an unmet medical need. Evidence of effectiveness is based on a surrogate endpoint or an intermediate clinical endpoint reasonably likely to predict clinical benefit. Applicants must verify and describe the anticipated clinical benefit in post-marketing confirmatory studies. Next slide. So here's what happens after accelerated approval. If the confirmatory study demonstrates the vaccine provides clinical benefit that clearly outweighs risk, then FDA grants traditional approval. If the confirmatory study does not demonstrate sufficient clinical benefit to justify the risks, then FDA has the regulatory procedures in place that could lead to removing the vaccine from the market. The key distinction between these two approval pathways is the level of acceptable residual uncertainty and how that residual uncertainty is evaluated, mitigated, and managed. Traditional approval essentially addresses uncertainty up front through persuasive and direct evidence of clinical benefit. Accelerated approval accepts greater residual uncertainty during the initial review, but mitigates and manages the acceptable residual uncertainty through required post-marketing confirmatory studies. To be clear, both approval pathways require substantial evidence of effectiveness for initial licensure. Next slide, please. Next slide, please. So let me define substantial evidence using our regulatory standard. It is evidence consisting of adequate and well-controlled investigations, including clinical investigations, conducted by experts qualified to evaluate effectiveness. The standard requires that experts qualified by scientific training and experience could fairly and responsibly conclude the vaccine will have the effect it claims to have under the conditions of use in the proposed label. Next slide, please. With the two approval pathways and that definition of substantial evidence in mind, Let's then turn to VRBPAC's charge today. VRBPAC is being asked to consider 3 points today. Next slide, please. First, do the primary endpoint, age subgroup, and strain-specific vaccine efficacy results from study P304 demonstrate clinically meaningful efficacy of Influenza against influenza disease in adults 50 through 64 years of age? Second, do the immunogenicity results from study P303 Part C comparing mRNA 1010 with Fluzone high-dose quadrivalent provide a reasonable basis to predict clinical benefit in adults 65 years of age and older? Please note that while immunogenicity is the primary evidence for inferring vaccine effectiveness in this age group, VRBPAC should consider the totality of available evidence, including, for example, the observed relative vaccine effectiveness compared with standard dose influenza vaccine in this population. And third, do the available data indicate that the safety profile of Influenza is adequately characterized, that identified risks are acceptable, and the residual risk can be appropriately monitored and managed through post-marketing pharmacovigilance? Next slide, please. So before turning the floor back to today's chair, I want to acknowledge the exceptional work that made this meeting possible. Thank you to FDA, CBER, OVR colleagues who prepared under an aggressive timeline for today's meeting. Thank you to all of today's presenters, including our public hearing contributors, for their input in today's VRBPAC discussion. And thank you to the VRBPAC members and today's chair for your expertise in today's topic and your dedication to protecting and advancing public health and enhancing public trust. Back to you, Dr. Monto.
Thank you, Dr. Kaslow. Next, I'd like to call on Dr. Joseph Kalinske of the Division of Review, Management, and Regulatory Review of CBER, who will speak to us about the biologics license application.
Hello, good morning. I'm Joseph Kalinske. I'm the review committee chair for review of the biological license application STN 125869. Next slide, please. Today I'm going to go over an intro for today. Start with a review of currently licensed influenza vaccines, go over some key features of mRNA vaccine technology, provide a high-level overview of the Influenza BLA, go over the agenda for the rest of our meeting today, and then end with the questions for the committee. Next slide, please. Okay, so this slide provides an overview of the seasonal influenza vaccines that are currently licensed and available for use in the United States. As you can see, there are several types available. You can see on the table on the left here that there are 6 currently licensed egg-based vaccines, where one is a high-dose formulation at 4 times the standard dose. One includes an adjuvant. As seen on the right, there are also licensed cell-based and recombinant vaccines, and one live attenuated vaccine. Each of these vaccines differs in terms of how they're manufactured, their age indication, and how they're administered. I'll also note that several vaccines are annotated as preferred options for specific age groups, and this is based on ACIP recommendations. In the interest of time, I'm not going to go through this in detail, but I'll pause briefly to allow the committee time to glance over the slide. Next slide. Okay, so while traditional cell and egg-based influenza vaccines have a decade-long safety record, have been demonstrated to be effective against influenza disease, and have been shown to reduce the risk of severe illness and hospitalizations, these vaccines generally require a 6 to 8 month production timeline from strain selection to first available supply. Furthermore, egg-based vaccines are susceptible to egg-adaptive mutations, and effectiveness varies year to year depending on strain match. mRNA vaccine technology allows for more rapid manufacturing, which carries the potential to facilitate a later, more precise seasonal influenza strain selection. These vaccines are also not subject to the propagation mutations associated with traditional cell and egg-based vaccines. Available data on seasonal influenza mRNA vaccines to date show generally robust immune responses, although there is some strain-specific variability, which is an issue that will be discussed more in depth in a presentation later on today. Also of note, in general, mRNA vaccines are associated with higher rates of transient local and systemic reactogenicity, such as injection site pain or fatigue, compared with cell or egg-based vaccines. And comparative effectiveness against high-dose or adjuvanted traditional vaccines will need to be evaluated further with time. Next slide. So the product being evaluated for licensure, mRNA 1010 has the accepted proprietary name of mFlusiva. You may hear the product being referred to as either mRNA 1010 or mFlusiva throughout the day, but I'll refer to it as mFlusiva for the purpose of this introduction. mFlusiva consists of a lipid nanoparticle, or LNP, encapsulated mRNA that encode hemagglutinin, or HA, glycoproteins of seasonal influenza strains. The product intended for licensure is a trivalent formulation that encodes the HA glycoproteins of influenza A strains H1N1 and H3N2, as well as Victoria lineage influenza B strain, with 12.5 micrograms of each mRNA, or 37.5 micrograms total mRNA per dose. The indication for mFlusiva proposed by the applicant is for active immunization for prevention of influenza disease caused by influenza virus subtypes A and B represented in the vaccine, and the intended target population is for adults 50 years of age and older. As Dr. Kaslow described previously, the regulatory pathway proposed by the applicant consists of a traditional approval for adults 50 through 64 years of age, supported by safety, immunogenicity, and clinical efficacy data, and accelerated approval for adults 65 years of age and older, supported by safety and immunogenicity data, relative vaccine efficacy data versus a standard dose comparator, and a post-marketing confirmatory study requirement. Next slide. Clinical efficacy and immunogenicity is supported in this application primarily by 2 phase 3 clinical trials, which you will hear about more in detail shortly from our expert clinical reviewers. In summary, data from Moderna's study P304 pivotal phase 3 primary efficacy study in individuals 50 years of age and older was included in this application to demonstrate that mFlusiva was non-inferior to the standard dose comparator in preventing influenza-like illness. Just to note, influenza-like illness is a set of symptoms that researchers can use as a standardized checklist to consistently identify and count potential influenza cases across trial participants, regardless of whether or not they have been formally diagnosed by a clinician. Immunogenicity data from Moderna's P303 Part C study was submitted to support the effectiveness of Influenza in adults 65 years of age and older, based on the use of hemagglutinin inhibition assay geometric mean titers and seroconversion rates as surrogate endpoints reasonably likely to predict clinical benefit. Both studies P304 and P303 Part C also serve as the two primary sources of safety evaluation for this application, each with approximately 6 months of follow-up. Moderna also conducted an integrated safety study, pooling data from our phase 3 studies to support a broader characterization of safety, which Dr. Brennan will cover in more detail later this morning. And so now to go over today's agenda following this introduction, next slide. We will start with a presentation from the CDC on seasonal influenza vaccine surveillance and effectiveness, followed by a presentation from our external speaker, Dr. Richard Webby from St. Jude, who will discuss the current state of seasonal influenza vaccines and some opportunities for improved effectiveness. We will then have a 10-minute break before we hear from Moderna. Next slide. And then following Moderna's presentation, next slide, FDA will detail the review of efficacy, immunogenicity, and safety of Influenza in adults 50 years of age and older. We will then break for lunch. Next slide. After lunch, we will begin our open public hearing, then followed by a 1-hour time slot for the committee to ask additional questions of the previous presenters. We will then have a 1-hour time slot reserved for additional committee discussion and voting. And with that, next slide. We have 2 voting questions we'd like the committee to consider today. 1, do the benefits of Influenza outweigh its risks for the prevention of influenza disease in adults 50 through 64 years of age? And 2, do the benefits of Influenza outweigh its risks for prevention of influenza disease in adults 65 years of age and older? That concludes my introduction, and I'm happy to take any questions.
Thank you, Dr. Kalinsky. Questions from the committee? Please unmute and let us see you. Dr. Perlman.
Yeah, so I had a question about the FDA document that was sent to us. There was a comment made about there being insufficient data about frail elderly individuals and immunocompromised individuals. And I wanted to, my question was basically, is this a standard part of vaccine approval, asking those questions? And second of all, especially for immunocompromised individuals, defining who should be in that category is difficult. So how is the FDA thinking about this?
I'll turn this over to Dr. Kaslow. Thank you.
Sorry. Thank you, Dr. Perlman, for that question. I think the intent is that we think it's important for special populations to generate evidence for special populations to help provide that evidence to healthcare providers and the customer of these vaccines. And so it's a signal, I think, to all of us actually that we need to generate this type of evidence. Is it required for licensure? No, it is not required for licensure, but it certainly would be nice to have that data. Now, it can be collected pre-licensure, post-licensure, but I do think it sends a signal as to the evidence gap, actually, in those special populations. So let me see if that answered your question, Dr. Perlman.
Yes, that mostly does. I'm partly just concerned about the definition of immunocompromised, because certainly for the SARS-CoV-2 vaccines, it was just tricky figuring out who actually is in that category, how much immunocompromised. And so doing the study is important, but it's hard to define who's in the appropriate categories.
I would say the office agrees with that assessment. It's complex, and I think we need to kind of figure out the optimal way of collecting that evidence. So agreed.
Okay, thank you.
Dr. El-Sadi.
Yes, thank you. So I have first a confirmation pertaining to the first population, 50 to 64. Dr. Caslow indicated that we are to examine strain-specific VE. I would like Dr. Caslow to confirm that this is our charge for today.
No, thank you for that question. I think the intent is for VRBPAC to look at the totality of the evidence, right? And those subgroup analyses, I think, provide a broader picture of the totality of the evidence. So that's the intent, including that in the points to consider by the committee.
Okay, because, strain-specific VE, if it is the main point of question 1, as you pointed out, this means we need formal statistical consideration for strain-specific VE. We'll get to that when we get to that. Now, the other...
So maybe I could just comment on that. I think the approach would be take a look at the primary endpoint from the pivotal efficacy study, focus on that. And then what we ask for in addition to one adequate, well-controlled trial is confirmatory evidence. Does the totality of the evidence support that the benefits clearly outweigh the risks? And so I think we wanted to flag that for the committee to consider the totality of the evidence.
Okay, thank you. Now, the other, more of a general question, and this pertains to the 65 and older population. You indicated that we are looking at an accelerated pathway that is to be approved in conjunction with a confirmatory post-marketing clinical study to confirm the VE in this population. If we look from a logistical and from a scientific approach, post-marketing studies always introduce bias. And if we are to avoid that bias, these studies will have to be even larger and more complicated. Wouldn't it be more cost-effective to consider pre-marketing clinical trials, especially that we have many options to safely immunize the population under question?
So good question again. I think I would defer that question until after the applicant has an opportunity to make their presentation. And in terms of cost effectiveness, I would actually direct that question to the applicant rather than to the FDA. After the applicant's response, if there's further questions, glad to address those from the FDA perspective.
Okay.
Thank you,
Dr. Caslow.
Dr. Berger.
Thank you. I have 2 questions. And the first one, I'm just going to ask FDA if you can provide us the regulatory definition of unmet medical need. I'm mostly just reflecting on the rationale that was provided, at least in terms of the mRNA vaccines. They seem to be more manufacturing concepts in terms of more precise strain selection at a later point in time. Does that meet the definition of unmet medical need from a regulatory standpoint? So if you could just give us a sense of what actually falls within scope, that would be appreciated. And then I have a second question.
I would say that piece of the equation probably is a little bit outside of scope of today's VRBPAC charge. It's certainly something that we're reviewing and discussing internally. Again, I would direct that question to the applicant for them to provide you how they're defining and looking at and making the case for unmet needs. So maybe come back to that question, Dr. Berger, after the presentations.
Will do. My second question is sort of coming off of what Dr. Perlman was raising with individuals who are frail or immunocompromised not being included in this study. I know, at least in the questions we're being asked to vote on, there doesn't seem to be a limitation in terms of intended use. It's just a totality. Do we approve for 50 to 64-year-olds? Do we approve for 65 plus? Generally, there would be the potential to restrict usage based on an intended use statement. Is that a possibility for consideration today, or are we limited to just the questions here?
Yeah, I would stay focused on the questions that have been proposed. A lot of the questions that you're asking, we actually deal with during our discussions with the applicant on the prescribing information and how it's characterized in the prescribing information. And there's a variety of different sections within the prescribing information where evidence gaps or uncertainty are highlighted. So, but I would say that is largely out of scope. That's something that we have a lot of work to do between now and the action due date as it relates to labeling.
Thank you.
Dr. Durbin.
Yes, I just had one comment with respect to the frail and vulnerable population, because in the demographics tables provided by FDA, 7% of participants 50 and older were frail and 18% were vulnerable. So my question is, I guess, did FDA mean that there wasn't breakdown in 64 and older? Because I did see those populations included in the phase 3 clinical trial.
Again, great question, Dr. Irwin. I will maybe defer that question until after my colleagues have made their presentation. I think they have lots of backup slides. I think they can address that question at that time, if that would be okay.
Sounds good.
Well, thank you, Dr. Kaslow and Dr. Kalinsky. We will next move to hear about the situation with influenza, seasonal influenza vaccine surveillance and effectiveness from Lisa Groskov, who I know very well, from CDC. Dr. Groskov.
Can you hear me?
Yes.
Excellent. Thanks so much, Dr.
Loud
Monta.
and clear.
Super. Thank you so much. Thank you very much. Good morning. This talk will provide an overview of U.S. influenza surveillance, as well as a summary of preliminary end-of-season influenza vaccine effectiveness estimates for the 2025-26 influenza season. Please note that the information presented today could change as more data are accumulated. Next slide. So we're going to start off with surveillance, part 1. The information we'll go over today for surveillance comes from the most recent CDC FluView report for the week ending June 6, 2026. Before I get started, one thing to note about this season, which we'll touch upon later, is that it was a drifted H3N2 season. Influenza H3N2 viruses predominated, with the majority of those H3N2s tested being from a genetically drifted subclade K. Next slide, please. So first, we're going to go over a couple of slides related to virologic surveillance. This slide summarizes influenza-positive test results reported to CDC by clinical laboratories in the United States. For orientation, percent of specimens positive is shown by the lines and the right-sided y-axis. Numbers of positive specimens divided by virus type, influenza A in yellow or B in green, is shown by the colored bars and the left y-axis. Surveillance week is on the x-axis, and that'll be the case for all the surveillance slides in this talk. So these data provide us with an index of influenza activity from the percent of specimens that were positive for flu, as well as the relative proportions of A and B viruses by week. The percent of specimens positive peaked for the season at over 30% during week 52, which was late December to the very beginning of January. Overall, considering the season as a whole, influenza A viruses, which are in yellow, predominated, and B viruses in green, had an increase in proportion on a week-by-week basis during the later weeks in the season. From the peak period at the end of the calendar year, moving right across the graph, we see that the percent of specimens positive has declined substantially in recent weeks, indicating low influenza activity at this time of year. Next slide, please. So this next slide summarizes virologic surveillance information also. This time it's influenza positive test results reported to CDC by public health laboratories. In contrast to what was shown in the clinical laboratory slide, in the last slide, the viruses isolated in the public health labs are more commonly tested for subtype for the influenza A viruses and lineage for influenza B viruses. So these data confer somewhat more granular data about circulating viruses by subtype and lineage as that information is available. Overall, for the season, we see that influenza AH3N2 viruses, which are shown in the figure in red, predominated. Among the H3N2 viruses, over 90% of those tested belong to subclade K, the aforementioned antigenically drifted influenza virus, which was originally identified in June 2025 after the selection of the 2025-26 vaccine viruses, and which differs from the AH3N2 influenza virus represented in the 2025-26 vaccine. Next slide. Next, we're going to change gears and move on to discuss outpatient-attended influenza-like illness, or ILI. These data come from the U.S. Outpatient Influenza-like Illness Surveillance Network, also called ILI-NET. One important thing to note is that ILI, influenza-like illness, is not a laboratory-confirmed influenza diagnosis. Rather, it's a symptom-defined entity defined as respiratory illness, including fever plus cough or sore throat. Therefore, illnesses caused by other viruses, such as SARS-CoV-2 and RSV, as well as others, can be captured in these data. However, the intensity of ILI activity generally temporally corresponds with influenza-specific surveillance data, such as the influenza-positive testing percentages presented earlier, as well as other indicators of laboratory-confirmed influenza activity. In addition, outpatient ILI activity is one of the indices used by CDC, along with estimated influenza-associated hospitalizations and deaths, to characterize the overall severity of an influenza season. This slide summarizes the percent of outpatient medical visits that were for ILI. So these are not rates, it's the percentage of visits that were for ILI, as reported weekly to CDC by approximately 3,000 outpatient facilities reporting to ILI-NET. For the current 2025-26 season, that's represented in red with superimposed circles. The 5 previous seasons for comparison are also shown. Similarly to the percent of influenza tests that were positive shown 2 slides back, the percent of outpatient visits reported to be for ILI peaked in late December. Next slide, please. This slide also describes data from ILI-NET, so it's showing the percent of visits that were for ILI, or influenza-like illness, by week, but this time it's only for the 25-26 season, and the data are broken out by age group. The percent of outpatient visits that were for ILI was the highest among the youngest age group, 0 through 4 years, followed by 5 through 24 years, 25 through 49 years, 50 through 64 years, and finally, those 65 years and older. Next slide. Changing gears again, we're going to discuss influenza-associated hospitalizations. This slide and the next one summarize laboratory-confirmed influenza-associated hospitalization rates. These data are from FluCert-Net, which is a system that includes 90 counties and 14 states and represents about 10% of the U.S. population. In contrast to the outpatient data on the last slide, these data are for hospitalizations rather than outpatient visits, and they're hospitalizations associated with laboratory-confirmed influenza. Rather than percentages, they show rates of hospitalization per 100,000 population. This figure shows the 25-26 season in red, as well as the preceding 4 influenza seasons. For the most recent 2025-26 season, hospitalization rates peaked in late December at 13.2 per 100,000. This rate is close to that observed for last season, 24-25, shown in blue, but occurred a little bit earlier in the season. You can also see that it's a somewhat narrower peak than we observed for 24-25. Next slide. These data are also from FluCert-Net, but we're focusing now on the 25-26 season. That's the only season on this slide. And this time we're breaking out the results by age group. Also different on this slide. It's still laboratory confirmed influenza-associated hospitalization rates, but I'm showing you the rates this time on a cumulative basis, not the week-to-week data that was presented in the previous slide. Since these are cumulative rates in the figure, the values are going to increase over time, reflecting accumulated hospitalizations over the course of the season. As is typical for most influenza seasons, the highest hospitalization rates were observed among those ages 65 and older, followed by those at the other end of the age spectrum, the youngest children aged 0 to 4 years old. The lowest rates were observed among older children, 5 through 17 years, and younger adults, 18 through 49 years. The overall cumulative rate across all age groups that you can see in this graph was, if you compare it to data back to the 2010-2011 season, the third highest that's been observed in that period of time. Looking a little more granularly at children in particular, it was the second highest since 2010-11 for children under 18 years. Even more granularly, second highest during that period for those 0 to 4, and highest for 5 through 17. Next slide, please. Our last two surveillance slides summarize mortality data. This first chart describes data from the National Center for Health Statistics Mortality Surveillance System. NCHS collects death certificate data for deaths occurring in the United States. The data shown here are for those deaths which are classified based on ICD-10 multiple cause of death codes as having been associated with influenza. As is the case for the previous slides, the surveillance week is on the x-axis. Percent of deaths reported to be due to influenza is on the left y-axis and is represented in the chart by the red line. Number of deaths is reported to be due to influenza is on the right y-axis and is represented in the chart in the orange fields. The number and percent of deaths coded for influenza for this season peaked in early January during week 53 and has declined to low levels since, paralleling the other trends in influenza activity we've seen today. Next slide, please. This is our last surveillance graphic, and it's for pediatric mortality. Pediatric deaths associated with laboratory-confirmed influenza have been nationally reportable since 2004. This slide depicts the number of deaths reported by week of death for the past 4 seasons, 2022-23 through 2025-26. For the 2025-26 season, as of June 6, 2026, 179 pediatric deaths have been reported to CDC. Notably, approximately 85% of children who were eligible for vaccination were not fully vaccinated against influenza. Next slide, please. To summarize the 2025-26 season based on the available data to date, seasonal influenza activity is currently low nationally. Overall for the season, influenza A virus have predominated, with influenza B viruses increasing and becoming a bit more predominant on a weekly basis later in the season. Among the influenza A viruses, H3N2 viruses predominated overall, with over 90% of those tested belonging to subclade K, the genetically drifted virus mentioned earlier. The cumulative influenza-associated hospitalization rate estimated by FluServNet is the third highest across age groups overall since the 2010-11 season. It was second highest since 2010-11 for those children under 18 years. A little more granularly, second highest during this period for the youngest children 0 to 4 years, and highest for 5 through 17 years. Overall season severity, based on estimated ILI outpatient visits and influenza-associated hospitalizations and deaths, is currently characterized as moderate across all age groups. It's characterized currently as moderate among adults 18 through 64 and those 65 and older, and high severity among children 0 through 17 years. 179 influenza-associated pediatric deaths have been reported thus far this season. Approximately 85% of those eligible for vaccination were not fully vaccinated against influenza. Next slide, please. Okay, moving on to part 2. This is the preliminary end-of-season estimates of vaccine effectiveness for 2025-26. Next slide. CDC's mid-season influenza vaccine effectiveness estimates from the networks that I'm going to discuss later were published in Morbidity and Mortality Weekly Report on March 12, 2026. The estimates we'll discuss today are preliminary end-of-season estimates. More data have accumulated since those estimates were published. These results are, however, again, preliminary and might change until the point where the data are finalized and everything finishes accumulating for the season. Next slide, please. To set the stage for this discussion, CDC estimates that there have been at least 32 million illnesses, 15 million medical visits, 390,000 hospitalizations, and 24,000 deaths due to influenza this season in the United States. Even a modestly effective influenza vaccine, therefore, has the potential to avert substantial burden in terms of absolute numbers. Next slide. The estimates we'll present today come from 3 separate CDC influenza vaccine surveillance network, influenza vaccine effectiveness networks, which evaluate vaccine effectiveness against laboratory-confirmed influenza for children, adolescents, and adults in both outpatient and inpatient settings. We'll describe the characteristics of these networks in the next few slides. Next slide, please. The 3 CDC influenza vaccine effectiveness networks we'll discuss today include the New Vaccine Surveillance Network, or NVSN, the U.S. Flu Vaccine Effectiveness Network, or US Flu VE, and the Virtual SARS-CoV-2 Influenza and Other Respiratory Viruses Network, or VISION. Next slide. The goal of these networks is to determine the effectiveness of seasonal influenza vaccination in the prevention of medically attended influenza, including influenza-associated outpatient visits, emergency department and urgent care visits, and hospitalizations. Next slide. Taken together, the data from these 3 networks include children, adolescents, and adults in both inpatient and outpatient settings. Here's a little bit more information on which age groups and outcomes are included in each network. The New Vaccine Surveillance Network, or NVSN, includes children, adolescents, 6 months through 17 years, presenting for outpatient visits, emergency department or urgent care visits, and hospitalizations. The U.S. Flu Vaccine Effectiveness Network, or US Flu VE, includes children and adolescents 8 months through 17 years. Because of the broad degree of overlap, you will see these data analyzed with the data for 6 months through 17 years when combined with other networks, and also includes adults 18 years and older, all presenting for outpatient, emergency department, and urgent care visits. So US Flu VE does not include hospitalization. The Virtual SARS-CoV-2 Influenza and Other Respiratory Viruses Network, or VISION, includes children and adolescents ages 6 months through 17 years and adults 18 years and older, presenting for emergency department and urgent care visits and hospitalizations. So once again, taken together, these data include information on children, adolescents, and adults in both outpatient and inpatient settings. Next slide, please. This map summarizes the geographic distribution of sites participating in the 3 networks. Taken together, they include patients from 16 U.S. states. Next slide. Summarizing the methods within the 3 networks. All enrollees are patients presenting for outpatient or inpatient care who have symptoms of an acute respiratory illness. The dates of enrollment for the data presented today are fall 2025 through spring 2026. The design used in all 3 networks is a test-negative case control design, comparing influenza vaccination odds among case patients with influenza confirmed by molecular assay versus control patients testing negative for influenza and SARS-CoV-2. Vaccination status is defined as receipt of any 2025-26 influenza vaccine according to medical records, immunization registries, claims data, and/or self-report. Next slide, please. For these analyses, vaccine effectiveness, or VE, is calculated as 1 minus the adjusted odds ratio times 100, expressed as a percentage. Estimates are adjusted for geographic region, age, and calendar time of illness. In addition, vision also adjusted for sex and race and ethnicity. US flu VE also adjusted for days between illness onset and enrollment, self-reported general health status, and sex. VE estimates were calculated for influenza A subtypes A, H1N1, PDM09, and AH3N2 when possible. However, influenza A subtype is not available in the data from a vision network. Lastly, VE is not estimated for some age groups and settings when the sample size was very small or confidence intervals very wide. Next slide, please. So we're next going to move on to discuss the preliminary end of season results, and we're going to start first with pediatric influenza vaccine effectiveness estimates from the 3 networks. Next slide. To provide a little bit of orientation, at the beginning of each age group section, you're going to see a table. This table reflects the proportion of influenza positive cases and influenza negative controls who were vaccinated in the second and third columns. The proportion vaccinated in controls is generally the best approximation that we have for vaccination rates in the general population. As you can see here, vaccination rates referred from that column are low, ranging from 28 to 47% across networks and settings. You also see in the last column estimated VE presented. We're actually going to discuss this information in a graphical format on the next slide. Next slide, please. For a little further orientation, throughout these slides, NVSN will be represented by the light blue horizontal line, US flu VE by the green horizontal line, and vision by a purple line. Within the horizontal lines, the shapes represent the point estimate for VE, and the horizontal lines themselves represent the 95% confidence intervals. Horizontal lines, that is confidence intervals, which cross the vertical dotted line, are not statistically significant. If the lines don't cross, they are statistically significant. In the example here, for example, you can see that for the US flu VE outpatient VE estimate, it crosses the dotted line and therefore was not statistically significant. So as for the data on this slide, among the different networks in outpatient settings, VE against any influenza in pediatric patients ranged from 11 to 46% in outpatient settings, and in inpatient settings ranged from 30% to 44%, apologies. Next slide. Next, we're going to go over influenza A. For influenza A overall, not subtyped, VE in outpatient settings ranged from 0 to 39%, and in inpatient settings ranged from 35 to 39%. Next slide. Next, some subtype-specific data. For influenza A, H1N1 PDM09, VE was 64% in the outpatient setting and was 48% and not statistically significant in the inpatient setting. Next slide. Now, influenza A H3N2. As noted earlier, the 25-26 season was characterized by a circulating AH3N2 influenza virus that was antigenically drifted, again referred to as subclade K. Estimated VE against H3N2 viruses in pediatric patients ranged from -1 to 34% in outpatient settings, and against hospitalization, it was 34%. Next slide. The 2025-26 season was also unique in that, in addition to the drifted H3N2 virus, a drifted influenza B virus also co-circulated. Among children, for influenza B, VE against influenza B ranged from 27% to 57% in outpatient settings and 20 to 51% in inpatient settings. Next slide, please. We're going to make another gear change and move to discussing vaccine effectiveness estimates among adults. Next slide. One thing just to remind people of NVSN includes only children, so the estimates you're going to see going forward are only coming from US flu VE and vision. Again, we see here in the third column the proportion vaccinated among the influenza negative controls, a approximation of vaccine coverage in the general population, which in this case for this age group, adults 18 and older, ranged from 39 to 47%. Next slide. First, this is data mirroring the last little group of slides. This is data for all influenza, any influenza, all A and B. VE ranged from 23 to 34% for outpatient visits and was 33% against hospitalization. Next slide. Against influenza A, VE ranged from 13 to 32% in outpatient settings and was 32% in inpatient settings. Next slide. Against influenza A, H1N1 PDM09, so we're moving into subtype-specific data, VE was 49% in the outpatient settings. An inpatient setting estimate is not available as there's no subtype-specific data available from Vision. Next slide. VE against H3N2 among adults was 8% in outpatient settings and was not statistically significant. Vision, again, doesn't collect subtype-specific data, so we don't have an inpatient estimate. Next slide. I believe the last slide in this series for adults, against influenza B. VE against influenza B ranged from 44 to 45%, and against hospitalizations, it was 45%. Next slide. The last thing we'll focus on in this part of the presentation is the subset of adults aged 65 years and older who are a group that are at increased risk for influenza-associated morbidity and mortality. Next slide. Vaccination rates, again, we're going to refer to the third column in this slide, prevalence of vaccination in the influenza negative controls, generally are higher for this age group than for other younger age groups. In this sample, they ranged from 54 to 66% across networks and settings. Next slide. These are estimates for all flu A and B. In outpatient settings, VE against any influenza for those 65 and older ranged from 31 to 37%. VE against hospitalization was 35%. Next slide. For influenza A, VE against outpatient visits ranged from 30 to 36%, and against hospitalizations was 35%. Next slide. VE against outpatient visits for H3N2 was 32% and not statistically significant. Vision, again, does not collect subtype-specific estimates. Next slide. For influenza B, VE against outpatient visits for influenza B was 52%. And for the inpatient segment, it was 43% against hospitalizations. Next slide. In summary, preliminary end-of-season analyses from the 3 CDC influenza B networks indicates that vaccination with 25-26 influenza vaccine reduced the risk for medically attended influenza outpatient visits and hospitalizations among children, adolescents, and adults, including older adults, across 16 U.S. states. Vaccination was effective against influenza A and B viruses and showed protection against H3N2. Vaccine effectiveness was lower than in recent previous seasons, notably 24-25, 23-24, but is consistent with broader trends in influenza B. Next slide. Thanks very much for your attention. In closing, I'd like to thank all of my influenza division colleagues, Alicia Budd and Patrick Maloney in particular, who provided slides, as well as those that work with them, also the influenza division staff, and all of the U.S. surveillance data contributors and the many collaborators from CDC, NVSN, U.S. Flu B, and Vision. Thanks very much. Happy to take any questions.
Thank you, Dr. Grosskopf. As usual, a very clear presentation. As Nancy Cox used to say, and we miss her very much, when you see one influenza season, you've seen one influenza season. We're talking pretty much in our pivotal study about the 2024-25 influenza season. Could you say a few words about that, in particular about the match between the circulating strains and the strains that were in the vaccine?
For the current season?
No, 24-25. That's what we're talking about in the pivotal study.
I see. There's a bit of a difference, obviously, between 2024-25 and the current season in that the advent of subclade K is something that we did not have during 2024-25. Overall, the VE estimates was somewhat higher for 24-25. However, this season, obviously, they're a bit lower. There's also, as you probably noted in the current season, some variability in the VE estimates in the different networks. As we know, the prevalence of different flu types and subtypes, as well as subclades, can vary geographically. If you want more specific information for the, more granular for the 24-25 season, I would be happy to pull that up and get that to the committee.
Okay, that may be useful. I blindsided you with that question, I know. But
I have a good idea, but I don't want to speak about specifics.
You're right. But it was a less drifted season than the current one.
Yeah, we have results this year that actually show promising effectiveness despite subclade K, but subclade K is a reality.
Okay, thank you. Dr. Gans.
Thank you so much for that presentation. I had a couple of more sort of granular questions, I guess, particularly about the younger age groups. First of all, I guess it would be nice to see vaccine rates in general, like how the vaccine is being used in all these different populations. And I think we have to start pulling out pregnant women, because that for us in the pediatric world is helping protect hopefully the very young. And that brings up that sort of population when we're looking at efficacy in particularly the young children, obviously, before they're able to get their own vaccines, how well that does. So it just, I think we need to start being more granular with our data polls and trying to subset that. So my first question is, what is the vaccine usage in general, and how is that changed? I know that's not going to change your vaccine efficacy data because you're just pulling people who have been vaccinated. But then to my second point, I just wondered if you had any data on the very young children and differences in that population in terms of their, when you went over the epidemiology. I think we need to subset out, again, our very young children so we can really understand different nuances in our vaccine usage. So those 0 to 6 months and in general under a year.
The estimates for coverage, likely, I believe, in general, are not finalized until the fall. There are preliminary estimates, which I can get for you. In general, one thing I can say that's been unfortunate over the last several seasons is that there has been a sort of a gradual stepwise decline in coverage since the advent of COVID pandemic. And that has affected actually all age groups, even those 65 and older. There's some variation because coverage actually is measured by a number of different instruments and surveys. But overall coverage has declined pretty much across age groups, even among the 65 and older, who have traditionally been the highest covered group. For specifics about the younger age of the spectrum and preliminary estimates, I can pull those for you for later. I think we have another Q&A session at the end of the day.
Thank you so much. I appreciate that.
Dr. Munoz.
Thank
Thank you very much, Dr. Gruxkopf. I had similar comment and question as Dr. Monto, but maybe to be more specific on the 65 and older, because I can see based on the ILI and the hospitalization rate data that 24, 25 and 25, 26 were actually quite similar in terms of the rates that we see. And this is almost going back to pre-pandemic era where we really see a high morbidity and hospitalization, especially in the 65 and up. So I was wondering if the fact that there's really almost no difference in that severity of flu in this last two seasons, even though we had drifted strains for the most recent one, what factors could be contributing to, again, not having been able to really have an impact. The distribution is always the same. 65 and older are the most affected, and the 0 to 4, and then the 50 to 65. And so that is very consistent. But I wanted to see if you had any comments specifically regarding the last two seasons, again, on coverage for the 65 and up specifically, and what effect that could have on VE estimates that we are seeing. Thank you.
Okay, thank you. I will gather that. Thank you, Dr. Newmans.
So we're going to be hearing some data about 24, 25 later on. Give you a chance to gather those data.
Thank you.
Seeing no additional raised hands, it's my pleasure to introduce Dr. Richard Webby of St. Jude Children's Research Hospital, who will be speaking on seasonal influenza vaccine, current status, and opportunities for improved effectiveness. Dr. Webby.
Thank you, Dr. Monto, and good morning, everyone. Perhaps we could just move on to the next slide. So I was tasked with providing a little bit of information on some of the topics that are relevant to perhaps some of the discussions in the context of where the current challenges in vaccine against influenza, particularly with regards to technologies we have. So three topics, basically: vaccine match, some examples of that, the role of egg adaptive mutations in vaccine candidate vaccine viruses, and then a slide on immune markers to follow up. So next slide, please. I think we're probably all fairly well aware of this, but just sort of to set the stage, the image on the slide shows from the University of Cambridge sort of a graphical representation of the antigenic evolution of, in this case, AH3 and 2 viruses from Hong Kong 68 when they moved into humans through into Fujian 02. And again, the distance between the dots here represents antigenic distance. And I think, as we know, particularly the H3 and 2 evolution is characterized by these spurts of change and antigenicity. Those change and antigenicity are driven, of course, by changes in the hemagglutinin molecule, and therein lies the issue we have against immunizing against influenza. That also happens to be the target for our current influenza vaccines. And that's really, from a virologic perspective, is allowed for by the fact that a lot of these major neutralizing epitopes on the influenza hemagglutinin are in flexible immunodominant loops, which can incorporate a number of amino acid changes while still maintaining the function of those proteins. And of course, that's really the driving force behind some of the global systems that are in place to monitor virus evolution, which, again, we've just heard some about. Next, please. So sort of heading to the match issue now. Obviously, not all seasons the vaccine that in use has a perfect match to circulating viruses. There are a number of reasons where this has occurred, but primarily limited to the challenges that we have in terms of both selection of strains, the timing of selection of strains relative to how long it takes to make influenza vaccines, get them vialed and get them distributed. And this particular image is showing, again, the influenza activity from FluNet, the WHO FluNet, just looking perhaps at the bars, the number of specimens tested, the peaks in the 2024 and 2025 influenza seasons. The challenge with this next slide, please, is that, take in mind the time needed to produce and distribute influenza vaccines. The recommendation, of course, for the next influenza season has to be made, at least Northern Hemisphere, has to be made in February of that year. And as you can see here, that's the arrow just highlights the point in terms the WHO, at least global recommendations for the Northern Hemisphere vaccine, at which time the Northern Hemisphere season has not finished, perhaps only just reached its peak, and the Southern Hemisphere season has not yet started. Next slide, please. I think a good example, which Dr. Grozkov just mentioned, is the current season, of course, was dominated by a drifted virus, the subclade K, H3 and 2. That particular variant wasn't actually detected until the Southern Hemisphere season and became quickly dominant and dominated that 2025 season. And clearly that the emergence of that particular virus was some weeks after the vaccine recommendations, and again, the vaccine recommendation driven by the timelines for influenza vaccine production and distribution. Next, please. We, I guess, also ask ourselves, so how much does that matter? So what evidence is there that match matters in terms of vaccine effectiveness? There's not a lot of information on this. The couple of examples I've given here on the left, here some investigators looked to quantify similarities against the antigenic region. So tried to correlate changes in antigenic sites that drive this mismatch, drive the antigenic evolution to vaccine effectiveness. And so the bottom line here is they develop models to translate simply the number of amino acid changes in key antigenic sites to vaccine effectiveness. And the picture at the bottom shows that. So it's got vaccine efficacy as taken from literature on the y-axis and sort of a measure of genetic similarity on key epitopes on the x-axis. Then you can see the two lines going down there, the dotted line represents the actual theoretical link between antigenic change, so match, if you like, and expected vaccine efficacy, and the solid line represents actually the observed vaccine effectiveness. So, at least some evidence there that yes, match does matter, and the similarity in these key antigenic regions does translate to vaccine effectiveness. On the right, a study from colleagues in Canada during the 2012-2013 season. This was a mismatch season. This particular mismatch was driven by egg adaptation, not necessarily change in the circulating viruses, but we'll come to that in the next slide. And in this study, they could show that certainly those egg adaptations were associated with antigenic change in the vaccine viruses, and looking across the different viruses that circulated in that season, there was a lower vaccine effectiveness against the H3 and 2, which, again, is not atypical, but again, suggestive of the fact that match does matter in terms of the effectiveness of influenza vaccines. Next, please. Just sort of a little bit of a word of caution on that. Of course, we did show in the last slide that there was a mismatch in the 2012-2013 season, and there was low vaccine effectiveness. But of course, what they didn't show that one directly led to the other. And this particular study here, utilizing the same data from the same season, In this particular case, using pre and post vaccination serum from people receiving the influenza vaccine. If we just look at the figure across the top panels, this measures these individuals' fold change, so the fold change in hemagglutination and inhibition titers from pre and post vaccination, measured against on the y-axis the actual vaccine virus IVR165, and against then the first panel across the x-axis Victoria 361, which was actually the virus from which IVR165 was derived, but without the egg adaptations, and then a couple of actual circulating viruses. And you can see sort of those lines across the top, fairly linear, suggesting that the vaccine effectiveness drop in the season perhaps wasn't due necessarily to the mismatch, that individuals vaccinated with this vaccine actually induced pretty comparable titers to the circulating strains as the vaccine strain. And here, at least in this case, they suggested that vaccine effectiveness was driven actually by lower immunogenicity of the antigen, not the mismatch itself. And of course, immunogenicity is not something that's available at the time of strain selection. Next, please. So moving on to the egg adaptation side of the problem. You know, it's been known for a long time, and this is to quote from a paper in 1987, that where they took samples directly from infected individuals, cultured virus from those individuals on both embryonated chicken eggs and also in cell culture, and they came up with the conclusion that the HA of virus cultivated in eggs was antigenically distinct from the HA of the virus from the same individual grown in mammalian cells. And again, some of the driver for the different platforms that are egg independent platforms that we do have. The reason for this is, of course, that we know a key part of the influenza virus replication cycle is attachment to the receptors on the surface of cells. Those receptors are slightly different in eggs than they are in humans and mammalian cells. The image down below again just shows on the structure of the hemagglutinin. In yellow is the key receptor binding domain. So again, the part of the molecule that engages the host receptor. And in red, we can see some key antigenic sites. And there's obviously a lot of overlap in between those. So as the virus changes its receptor to adapt more efficiently for replication in eggs, it changes that receptor binding domain. As there are overlap between that domain and key antigenic sites, sometimes those also drive antigenic change in the virus as well. Next, please. So the egg adaptation has been sort of a recurring, but I'd say somewhat unpredictable property in influenza vaccine strain selection and mismatch. I'd say unpredictability, both in terms of when it arises, but also in terms of the magnitude of impact it has. The H3 and 2 viruses are often the problem in terms of the egg adaptation, but it's not limited to that. Here's a slide actually that was presented at the March VRBPAC meeting to support the strain recommendations for the upcoming influenza season. Highlighted on the left-hand panel in blue is HI data from cell antiserum generated against cell Pennsylvania, homologous titer of 80. In the blue box there, that serum also recognizes anticipated virus to circulating the next season very well. If we look on the next box, the egg Tokyo is essentially the egg-grown version of cell Pennsylvania, and there we can see homologous titers of 1280 and slightly lower titers to circulating viruses. So in this case, the bees are also affected by egg adaptation, in this case, a loss of a glycosylation site upon adaptation to eggs. Next, please. Of course, adaptations are fine, they do happen, but they are also targeted by the human immune response. A number of studies have looked at this. Here's just a summary of a study from the US CDC, again looking at polyclonal responses to H3 and 2 viruses from the 2016 to 2019 seasons, when there was a spate of egg adaptations in the H3 and 2 viruses. perhaps we could just look at the graphic for now. They did clearly see targeting of these egg adaptations, but it was age dependent, probably impacted previous natural infection vaccination. In these studies, young children who had only been, the first exposure was the vaccine itself, they had quite a targeting of their antibody response against these egg adaptations. Young children who had previous natural infection followed by this vaccination weren't quite as skewed towards egg adaptation. And in the elderly here, these are elderly individuals who had been vaccinated repeatedly with vaccines that contain these egg adaptations. Again, there was a good percentage of the polyclonal response seen to be targeted against the egg adaptations. So egg adaptations do occur. They do occur with some frequency, and they do seem to impact the host response. So next, please. So sort of just a final slide for the historical Hobson study, different studies from human challenge studies and neutralizing activity of antibodies, serum antibodies. HI of 18 to 32 provided 50% protection, and that sort of HI of equal or greater than 40 has held true across a number of natural infection studies, also inactivated vaccine studies as well. The HI protection has not been formally demonstrated across all vaccine platforms, although the expectations are that it would hold, at least for those that target the hemagglutinin as sort of the active ingredient of the vaccine, if you like. I think particularly as we have seen this HI 40 across the natural infection studies as well, where perhaps a range of immune responses are being employed. I think there's a little bit of clarification or lack of clarification on what exactly the HI 40 is. Is it actually a correlate, or is it actually an actual measure of protective mechanism? So certainly we think that neutralizing antibodies that target HA, of which HI is a surrogate, are the major driver of protection. And so it's perhaps the strict definition of correlate, may not necessarily be the most appropriate here, and perhaps it's an actual measure. We could argue, of course, this is measured in the serum, so the periphery of people, where, of course, the action is really the mucosal surface. But those are discussions. So I think that's my last slide. I thank you all for listening. Oh no, unmet needs. Sorry. Yes. So what does that all mean? I think so. Where we need to go with influenza vaccines, we need additional platforms that offer more flexibility in the timelines. Perhaps their manufacturing process is faster, so those decisions can be delayed a little bit. Gives us a little bit more time, perhaps, to get through the rest of the Northern Hemisphere season before decisions need to be made. Platforms that are less prone to adaptive mutation. So whether that be egg adaptations, of course, we do have egg-independent platforms now. At least one of them is recombinant protein expressed in insect cells. But even with that, it's these are insect cells. The HA is glycosylated. It's a little unclear about the difference in glycosylation between insect cells and mammalian cells and that impact. But certainly, platforms that don't drive adaptive changes. It's also important that the next point is really the scalability. So platforms that do those first 2 points but don't manufacture in the scale needed probably have minimal impact. And then, of course, we all want to get to eventually is vaccines that target multiple immunogens, multiple parts, stimulate multiple parts of the immune system, or more conserved antigens. And so now I'm finished, and thank you.
Thank you, Dr. Reddy. Would you agree that based on available evidence that we know that antigenic match matters, but we still really can't predict very well how much?
Yes, I would absolutely agree with that. Yes. And again, the point there, in terms of vaccine whatever vaccine effectiveness, efficacy, I think there are multiple things that do play into that match just being one.
Yeah, and it isn't just the antigenic match.
No, it is not.
Okay, thank you. Dr. Gans.
Thank you so much once again for that excellent presentation. Sort of related to what Dr. Monto was just asking, I think the impact of the egg changes, so the egg adaptation, how much does that actually affect the match versus not match question? I guess, is that also impacting the just overall efficacy of the ability of these vaccines to really manufacture a product that actually, quote, matches, since it's human versus something that was having these adaptations to grow in that. And then like the follow-up question to that is, then how does the non-egg based, it's glycosylated in insect, how does that variation, the end product, then match, again, the human strain? So those are strain related. And then I did have a question because you went over very quickly. My third question is related to the immune response. And unfortunately, I think we didn't have these slides updated in our box, but so I didn't get to look at this in advance. But you made a suggestion that there was an impact on your ability to make future responses when you've seen these egg-derived whatever. And I wondered about that because, of course, you're going to have an immune response to the proteins that you see, which is it your opinion then you don't broaden your immune response when you see different antigens? Because I would just expect those to stay, but you're able to broaden out your ability to have a breadth of their immune response. So that's my third question. Thank you.
Okay, certainly. So correct me if I don't get all these questions right here. The first, sort of that real impact on egg adaptation versus match. I would say in the last, couple of decades, a fair percentage of the mismatch seasons have been driven by egg adaptations, as opposed to sort of true changes in the antigenicity of circulating viruses. Again, the relative impact on that on vaccine efficacy probably varies from season to season. But so I would, these in terms of delayed emergence of a variant versus egg adaptations are again probably on equal footing in terms of driving that mismatch problem. Until sort of the comment on the recombinant, product and grown in the glycosylation difference, I don't think we know. Certainly, at least I'm unaware of data that suggests the impact of that. It may have an impact, it may not. And last, sort of the issue of sort of how different exposures broaden or not the immune response. I think we're starting to understand a lot that those very first exposures to influenza antigens do influence how you respond to subsequent exposure. So they do sort of the imprinting, original antigenic sin issues where, those first key exposures do actually have a major impact on how you respond later on. So again, it's not that these can't be broken. So again, if the first exposure, the two kids in this case, the example I gave, those that received the vaccine first, again, targeted these egg adaptation in a pretty key and immunodominant part of the virus. But those kids that have been primed with an infection with viruses that didn't have those changes, their response to vaccine was more targeted against the shared epitopes between the original infecting strain and the vaccinating strain, which in this case was not the egg adaptation. So the broadening of response, yes, probably does happen upon repeated exposure, but it's also very, very dependent on what the original exposure is.
I guess we do have to sort of really dig into that because it will be different. If you have one antigen, you're going to make an immune response to that. What my question is it really shouldn't impact your ability to then further see different you may boost that one more than other ones that you're seeing, but you still should have the ability to make antibody responses specific to other natural virus epitopes. So I think because when we use the terms, imprinting this and that, it suggests that then your immune response that follows that, while different because you've seen different antigens, is actually less. So I think that's where I think we just have to be very careful. If that's true and we think that's less efficacious, that's a different story than we have a different profile. Having a different profile just means what you've seen, and your immune system's very specific. So I guess that's what I'm really trying to get at and sort of really hone in on, because people seem to carry this forward as something that is, quote, bad.
Right. Yeah, that point taken. And again, I think there's a lot of variables within that as well, of course, in terms of what, the natural exposure downstream that they're sort of faced with maybe as well, and how that differs or not between previous exposures. But no, the point taken. It doesn't negate your ability to change that response over time with repeated exposures to perhaps improved and real antigens. Yeah.
I would suggest that everybody review the original paper from Dr. Francis, written in the 1950s about original antigenic sin, because he concludes that the answer to original antigenic sin is vaccination, to bring things more up to date. So, Dr. Webby, thank you so much for your presentation about our gaps.
You're very welcome. Thank you.
And this brings us to our 10-minute break. So we will return in about 8 or 10 minutes. I won't give you the exact Eastern time, but it's going to be up on your screen, I'm sure.
You may begin.
Welcome back to the 193rd meeting of the VRBPAC. Next, we will be hearing an hour's presentation from the sponsor. The title of the session is Moderna Investigational Influenza Vaccine mRNA 1010 in adults greater than 50 years of age. Dr. Das, will you introduce your colleagues as they go down through the presentation?
Yes, Dr. Monto, I will.
All right, thank you very much. And you're in charge of the overall presentation.
Great, thank you very much. Good morning. My name is Rita Das, and I'm Senior Vice President of Clinical Development in Infectious and Rare Diseases at Moderna. I'm an adult, pediatric, and infectious diseases physician by training, and I previously led Moderna's COVID and RSV portfolios. Thank you for the opportunity to present Moderna's clinical data supporting approval of mRNA 1010 for the prevention of influenza in adults 50 years of age and older. mRNA 1010 is Moderna's mRNA-based seasonal influenza vaccine candidate. It encodes membrane-bound hemagglutinin antigens of WHO-recommended seasonal influenza strains and does not require propagation in eggs or other complex cell culture systems. This is important because current influenza vaccine technologies can be affected by the long interval between strain selection and vaccination, as well as by egg-adaptive changes that may alter the vaccine design. By directly encoding the selected antigen sequence, avoiding egg adaptation, and supporting a more flexible manufacturing timeline, mRNA 1010 has the potential to address important limitations of current influenza vaccines and improve protection in a population that continues to experience substantial influenza disease burden. Moderna is seeking approval of mRNA 1010 for adults 50 years and older. The proposed vaccine is administered in a single intramuscular dose in a prefilled syringe. The application was structured using two regulatory pathways: traditional approval for adults 50 to 64 years of age and accelerated approval for adults 65 years and older. The central clinical question before us today is whether the totality of data supports the approval of mRNA 1010 for adults 50 years of age and older, and we will present the data in that context. Our clinical program for mRNA 1010 was designed to support approval in adults 50 years of age and older. It was developed through multiple FDA interactions, including discussion of the pivotal efficacy study design and licensed standard dose comparator. In alignment with FDA's feedback and consistent with previous studies of enhanced influenza vaccines, licensed standard dose vaccine was used as a comparator for all adults 50 and older in the efficacy study. After the BLA submission, FDA highlighted different ACIP recommendation for influenza vaccines in adults 65 plus. To address FDA's question about the comparator, we proposed a modified regulatory framework at a Type A meeting. For adults 50 to 64 years of age, Moderna is seeking traditional approval based on direct clinical efficacy versus a licensed standard dose vaccine, supportive immunogenicity, and an acceptable safety profile. For adults 65 and older, Moderna is seeking accelerated approval based on superior immunogenicity data versus a high-dose vaccine, direct clinical efficacy against standard dose vaccine, and an acceptable safety profile, with confirmatory effectiveness data against an enhanced influenza vaccine to be provided post-licensure. Additionally, approval in both groups is supported by the benefits of the mRNA platform for influenza vaccines. Influenza remains a serious and often life-threatening illness. In adults 50 years and older in the United States, seasonal influenza continues to result in substantial numbers of medically attended illnesses and hospitalizations each year. Across recent seasons, CDC estimates show that adults in this age range experience millions of medically attended influenza-related events and hundreds of thousands of hospitalizations annually. This ongoing burden underscores that despite the availability of vaccines, influenza prevention remains an important public health need in adults 50 years and older. The burden of influenza is especially pronounced in older adults. In the 24-25 influenza season, in the U.S. adults 50 and older, CDC estimated more than 546,000 influenza-related hospitalizations and 41,000 influenza-related deaths. Adults 65 and older accounted for a majority of these severe outcomes, approximately 57% of all influenza hospitalizations and 71% of deaths. This reflects the well-established vulnerability of older adults who are more likely to have underlying medical conditions and more likely to experience complications with influenza. Together, these data highlight the need for improved influenza prevention, particularly among adults at highest risk for severe disease. This need is reflected in ACIP's preferential recommendation for enhanced influenza vaccines in these populations. Licensed influenza vaccines remain an important foundation of public health, and at the same time, current technologies have known limitations. First, strain selection occurs months before vaccination season. During that interval, circulating viruses can continue to evolve. Second, egg-based manufacturing can introduce egg-adaptive changes that alter the vaccine design. This is especially relevant because commercial data indicate that approximately 95% of influenza vaccine doses administered in the U.S. to adults 65 and older are egg-based. Together, long manufacturing timelines, viral evolution, and egg adaptation can contribute to strain mismatch. They also highlight an important public health priority, diversifying influenza vaccine manufacturing beyond egg-based approaches. Licensure of mRNA 1010 would also provide a seasonal influenza vaccine platform to support influenza pandemic preparedness. To put the limitations of strain mismatch into context, a recent review of Northern Hemisphere influenza season from 2011 through 2020 found that antigenic drift contributed to mismatch in 4 of 9 seasons, while egg adaptive mutations contributed to mismatch in 6 of 9 seasons. Across that period, mismatch from drift or egg adaptation was documented in all but a single influenza season. When the vaccine antigen differs from the circulating virus, vaccine effectiveness can be reduced. This is one reason why a platform that can preserve antigenic fidelity and potentially allow later strain selection is so relevant for seasonal influenza. The clinical consequence of these limitations is reflected in the variability of seasonal influenza vaccine effectiveness over the last decade. Over this time, CDC estimates show that influenza vaccine effectiveness against medically attended illnesses has varied substantially from season to season. Some seasons show very meaningful protection, others show limited effectiveness. The important point is not that current influenza vaccines lack value. They clearly remain an important public health tool, but rather their performance is variable, and influenza continues to cause substantial morbidity and mortality despite vaccination. This ongoing burden supports the need for improved vaccines. The mRNA 1010 clinical development program provides efficacy, immunogenicity, and safety data across more than 71,000 adults in the intended use population. The pivotal efficacy study, Study 304, evaluated mRNA 1010 against a licensed standard dose influenza vaccine in more than 40,000 adults 50 years of age and older. Study 303C evaluated immunogenicity and safety of mRNA 1010 versus a licensed high-dose influenza vaccine in almost 3,000 adults 65 years of age and older. In addition, we have prepared an integrated summary of safety across 4 phase 3 studies, including more than 71,000 adults 50 years of age and older. This includes evidence from 2 earlier phase 3 studies, 301 and 302, These studies contribute relevant safety information, although they evaluated a prior version of the vaccine that was subsequently discontinued in favor of the optimized candidate that's being discussed today. Today, we will show that the totality of evidence supports a positive benefit-risk profile for mRNA 1010 in all adults 50 years and older. The pivotal efficacy study demonstrated superior efficacy compared with a standard dose influenza vaccine, with consistent protection across strains, high-risk groups, and age subgroups. The immunogenicity data show higher immune responses compared to both standard dose and high-dose influenza vaccines. The safety data show that local and systemic reactions were more frequent with mRNA 1010 than with comparators. They were generally mild to moderate, short in duration, and resolved without medical attention. Importantly, no safety concerns were identified. Taken together, these data support the potential for mRNA 1010 to reduce influenza burden beyond currently licensed vaccines. The regulatory framework under which influenza vaccines are licensed is based on the demonstration of safety and effectiveness in the intended use population. No currently licensed influenza vaccine carries a separate indication of being an enhanced vaccine. Rather, enhanced vaccines demonstrated added benefit relative to standard dose vaccines, and subsequent recommendations evolved based on the strength of those data. Consistent with that framework, the evidence supporting mRNA 1010 is aligned with the evidence used to license other enhanced influenza vaccines. For Fluzone High Dose and Flublok, licensure was supported by demonstrations of added clinical benefit relative to standard dose influenza vaccines, with relative vaccine efficacy estimates ranging from 24 to 30%. Fluad was licensed based on immunogenicity data without direct efficacy data. In Study 304, mRNA 1010 demonstrated relative vaccine efficacy of 26.6% in adults 50 years and older and 27.4% in adults 65 years and older. The lower bounds of the 95% confidence intervals were 16.7 and 12.1% respectively, numerically higher than those reported in the pivotal efficacy evaluations for Fluzone High Dose and Flublok. Taken together, both the magnitude and precision of mRNA 1010 efficacy results are consistent with the evidence used to support licensure of other enhanced influenza vaccines. Here's the agenda for the remainder of today's presentation. My colleagues and I will first review the burden of influenza in the United States and the effectiveness of current influenza vaccines. We will then present the efficacy, immunogenicity, and safety data from the mRNA 1010 clinical program. I will return at the end of the presentation to summarize the data and to moderate today's question and answer session. I will now turn the presentation over to Dr. Evan Anderson.
Thank you, Dr. Doss, and good morning. My name is Evan Anderson, and I'm Vice President of Epidemiology at Moderna. Prior to joining Moderna, I was a practicing infectious disease physician and influenza clinical researcher at Emory University. I will review the clinical burden of seasonal influenza in adults 50 years of age and older, and discuss how factors such as viral evolution and manufacturing timelines contribute to the performance of currently licensed influenza vaccines. As Dr. Doss introduced, influenza remains a serious and potentially life-threatening illness in adults 50 years of age and older, and the impact goes beyond the acute respiratory infection. Influenza can trigger clinically important complications, particularly in older adults and in people with underlying medical conditions. These events include acute cardiovascular events such as myocardial infarction, heart failure, and stroke. Influenza can also lead to pulmonary complications, including pneumonia and exacerbations of chronic obstructive pulmonary disease. And it has been associated with neurologic, muscular, and renal complications. Together, these complications contribute meaningfully to the overall clinical burden of influenza. The risk of serious influenza-related outcomes increases sharply with age, with the greatest burden in adults 65 years of age and older. This slide shows CDC data from the 24-25 season. Compared with adults 18 to 49 years of age, adults 50 to 64 had nearly a 3-fold higher rate of influenza-associated hospitalization and more than 7-fold higher rate of death. The risk was even higher in adults 65 years of age and older, with more than an 8-fold higher rate of hospitalization and more than a 25-fold higher rate of death compared with young adults. Those over the age of 50 account for over 3/4 of hospitalizations and over 90% of deaths. These data make clear that severe influenza burden is concentrated in adults over 50, and particularly among those 65 and older. During the 24-25 season, the season of the pivotal mRNA 1010 efficacy trial, influenza A viruses, shown in blue, predominated. Influenza A, H1 and H3N2, circulated at similar levels and peaked in January. Influenza B, shown in orange, circulated at lower levels overall and peaked in March. This pattern is important when interpreting the clinical data. Influenza B remains an important component of seasonal influenza vaccines. However, it has historically represented a smaller proportion of cases in older adults. Influenza A has been the predominant driver of seasonal influenza. Over 9 seasons, influenza A accounted for over 83% of influenza-related hospitalizations in adults over 50. The 24-25 season was similar in that influenza B accounted for less than 1% of cases in adults 65 years and older among specimens sequenced in U.S. public health laboratories. Today, seasonal influenza vaccines are made using several different technologies. Enhanced vaccines, which include high-dose, adjuvanted, and recombinant influenza vaccines, are recommended for adults 65 years of age and older in the U.S. There are 2 key limitations of current influenza technologies. First, with egg-based vaccines, the virus can change as it adapts to grow in eggs. These egg adaptive changes can alter the vaccine antigen from the strain that was originally selected. Second, influenza viruses continue to evolve over time. Current influenza vaccines must be selected and manufactured months before we know exactly which viruses will circulate. Antigenic drift can occur after strain selection and before the influenza season. These 2 mechanisms contribute to the mismatch between vaccine strains and circulating viruses. The schematic shows why timing is such an important challenge for seasonal influenza vaccines. For fall vaccination campaigns in the U.S., vaccine strains are selected months before vaccines are administered. After strain selection, manufacturers need time for seed and manufacturing readiness, production, quality testing, and distribution before the vaccine season begins. This timeline is essential to ensure broad annual vaccine availability. But during this approximately 6-month window, influenza viruses will continue to evolve. The COVID-19 vaccine annual strain update process provides a useful example of the flexibility of the mRNA platform. For COVID-19, strain recommendations are made much closer to the vaccination season, usually at the end of May, with updated mRNA vaccines available for the fall season. While influenza and COVID-19 are different programs, this illustrates an important platform advantage. Once the strain is selected, the vaccine can be rapidly updated. The 25-26 Northern Hemisphere season highlights the potential advantages of shortening the manufacturing timeline. WHO strain selection occurred in February 25. After strain selection, an AH3N2 subclade referred to as subclade K emerged in June of 25. This subclade contained hemagglutinin mutations that affected recognition by vaccine-induced antibodies. During the US season, subclade K accounted for almost 93% of circulating H3N2. This illustrates the practical consequence of the long interval between strain selection and vaccination. Influenza viruses continue to evolve after making vaccine composition decisions. Across the past 15 influenza seasons, CDC reported vaccine effectiveness has varied substantially among adults 65 years of age and older, from as low as 12% up to 50% against outpatient medically attended infections. Evidence of egg adaptation, symbolized by the egg, or antigenic drift, symbolized by the blue virus, have been documented in at least 13 of the past Northern Hemisphere influenza seasons. In the 24-25 season, the time of the pivotal study, there was a partial H3N2 mismatch. Although egg adaptation and antigenic drift do not explain all the variability observed in vaccine effectiveness, they do highlight persistent vulnerabilities of currently licensed influenza vaccines. Substantial room exists for improving influenza vaccine effectiveness. Finally, as Dr. Webby nicely alluded to, there is a potential type B mismatch in the upcoming 26-27 influenza season for egg-based vaccines. Influenza B Victoria subclade C3.1 increased in prevalence in the US during the 25-26 influenza season. This new clade has multiple mutations that resulted in antigenic escape. As a result, the 26-27 Northern Hemisphere influenza B vaccine composition was updated to an influenza B Victoria subclade C3.1 strain. B Tokyo was selected for the egg-based vaccines due to its ability to grow in eggs, but egg adaptive mutations resulted in loss of a glycosylation site. For non-egg-based vaccines, the recommended strain is B Pennsylvania. This strain includes a glycan present at site 197 of the hemagglutinin antigen that cannot be replicated in eggs. Early immunogenicity testing has shown an approximately 8-fold reduction in HAI titers with the egg-based strain compared to the non-egg-based strain against circulating influenza B Victoria C3.1 viruses. Although the clinical relevance is not currently known, this highlights the potential value of an mRNA-based vaccine for improving strain match and immunogenicity. In summary, influenza remains a serious and potentially life-threatening illness in adults 50 years of age and older, with the greatest risk in adults over the age of 65. Influenza also contributes to serious cardiovascular, pulmonary, and other complications, adding to the overall clinical burden in older adults. Influenza A is the primary driver of severe influenza-related outcomes among older adults. Currently licensed influenza vaccines remain an important foundation of public health. However, vaccine effectiveness varies across seasons and can be affected by lengthy manufacturing timelines, antigenic drift, and egg adaptation. Together, these challenges highlight the need for improved influenza vaccines that can help reduce morbidity and mortality in older adults and better address the limitations of current seasonal influenza vaccine technologies. For adults 50 years of age and older, there remains an unmet need for improved influenza prevention. I will now turn the presentation back to Dr. Das.
Thank you, Dr. Anderson. I will now discuss the clinical efficacy and immunogenicity data for mRNA 1010. There are 3 key studies for the development program. The first is the phase 1/2 study, Study 101. This evaluated several doses and determined that the 12.5 microgram per strain dose provided the ideal balance of tolerability and immunogenicity. This informed the dose selection for our phase 3 studies, and the details are provided in our briefing document. Next, our phase 3 relative vaccine efficacy trial, Study 304, where more than 40,000 adults over the age of 50 were randomized to either mRNA 1010 or standard dose influenza vaccine comparator. In this study, we demonstrated superior vaccine efficacy compared to the comparator. And finally, the phase 3 study 303 Part C, in which nearly 3,000 participants 65 and older were randomized to receive either mRNA 1010 or high-dose influenza vaccine. In that study, we demonstrated superior immunogenicity compared to high-dose influenza vaccine. During the clinical development program, mRNA 1010 transitioned from quadrivalent to a trivalent formulation in line with WHO and FDA strain recommendations. While Study 303C evaluated the earlier quadrivalent formulation, each strain was included at a 12.5 microgram dose. In Study 304, we evaluated the current trivalent formulation for licensure, and the per strain dose did not change. Now I'll present the results of our pivotal vaccine efficacy trial. Study 304 was a randomized, double-blind, active controlled study designed to evaluate the relative vaccine efficacy of mRNA 1010 as a trivalent formulation to standard dose influenza vaccine. Both trivalent and quadrivalent vaccines were included in the active control group based on vaccine recommendation in the geographic region. In general, the U.S. had transitioned, but some of the European geographies in which the study was conducted had not yet. Over 40,800 participants were enrolled and randomized 1-to-1. Participants were stratified by age, 50 to 64 and 65 years and older, to ensure good representation of older adults. Influenza vaccine status for the previous influenza season was captured. Because Study 304 accrued endpoint cases rapidly in the high severity 24-25 influenza season, a total of 968 cases accrued, the target case count for the study, which was 836, was exceeded in that single season. There are several reasons why a licensed standard dose influenza vaccine was considered an appropriate comparator for the efficacy study. The comparator strategy was prospectively discussed with the FDA during program development and aligns with the comparator approaches used in the development programs for enhanced influenza vaccines. This is also consistent with regulatory guidance for influenza vaccines from FDA and ICH. Additionally, Study 304 was conducted across multiple countries where recommendations for enhanced influenza vaccines differ. In some of these countries, standard dose influenza vaccines are widely used in all age groups. Finally, the phase 3 program directly addressed the enhanced vaccine question in Study 303C through immunogenicity comparisons with high-dose influenza vaccine in adults 65 years and older. The intent in our development program was always to evaluate mRNA 1010 effectiveness compared to enhanced influenza vaccines, but post-licensure. The primary objective of the study was to demonstrate non-inferiority and then superiority of the relative vaccine efficacy of mRNA 1010 versus the standard dose influenza vaccine, as well as to assess vaccine safety and reactogenicity. Secondary endpoints included immunogenicity in a subset of 2,400 participants. A key exploratory endpoint was to evaluate the relative vaccine efficacy of mRNA 1010 for medically attended influenza, where the greatest burden of flu morbidity and mortality occurs, particularly in older people. First, I'll present the study demographics. The demographics and baseline characteristics of the study population were well balanced between groups by age, sex, ethnicity, and previous vaccination status. 70% of participants were from North America. Frailty was assessed in those over 65, and over 5,000 participants, or 27% of those over 65, were classified as frail. Using the protocol definition, 57% of participants had a baseline high-risk condition. This included obesity, diabetes, asthma, and chronic obstructive pulmonary disease. Using an alternate high-risk definition, which incorporates additional conditions that may increase influenza risk, including arthritis, previous cancers, depression, hypertension, and hyperlipidemia, 82% of participants were considered at high risk. Participants with other immunocompromise, such as transplant, that may impact their response to vaccines were not included in the efficacy study. Study 304 was a large, multinational, pivotal efficacy study conducted at more than 300 sites across 11 countries in the Northern Hemisphere. The U.S. had the largest footprint, and more than 27,000 participants were enrolled in the U.S. Local ethics committees and national health agencies provided oversight and approval of the study in each country. In addition, the entire study was overseen by an independent external Data Safety Monitoring Board, or DSMB, which could review unblinded safety data if needed and made the recommendation to complete the study based on success at the end of the 24-25 season. Now I will share the study methods. We conducted active influenza surveillance throughout the duration of the Northern Hemisphere influenza season. An electronic diary with twice-weekly prompts was used to collect any respiratory symptoms. If symptoms were reported, the site scheduled a clinical assessment within 72 hours, and a nasal swab was collected for central laboratory testing. Participants were also instructed to measure their temperature daily during episodes and notify study personnel for any temperature over 37.2 degrees Celsius. Any events resulting in medical attention, including clinic, urgent care, emergency room visits, or hospitalizations, were reviewed to determine if these events were associated with an influenza diagnosis. Here are the fuller case definitions for influenza-like illness used in this trial. As a reminder, all definitions did require RT-PCR confirmation of influenza within 7 days of illness onset. The first definition shown here was used in the primary analysis of relative vaccine efficacy. The CDC and WHO definitions can be used as an indicator for more severe disease as they required fever. Now I will share the relative vaccine efficacy results. As a reminder, relative vaccine efficacy describes the improvement in efficacy of mRNA 1010 over the standard dose vaccine. In this study, 3 pre-specified statistical tests to assess relative vaccine efficacy were performed in sequence. First, non-inferiority with a lower bound of the confidence interval greater than -10%. Then superiority with a lower bound of the confidence interval of 0%. And finally, the most stringent test, highest superiority, with a lower bound of the confidence interval of 9.1%, which was the same superiority criteria used in the Fluzone HD efficacy trial. In our study, mRNA 1010 demonstrated an increased vaccine efficacy of 26.6% over the comparator vaccine. These results met all pre-specified success criteria, including both non-inferiority and both levels of superiority. Here you can see that our lower bound of the 95% confidence interval was 16.7, and the p-value for this comparison was 0.0005. Relative vaccine efficacy was consistent and significant across all case definitions. For the WHO definition, with the highest level of fever, the RVE was 29.7%. In evaluating protection against the individual influenza strains in the vaccine, we demonstrated consistent relative vaccine efficacy across all 3 strains, with the RVE of 29.6 against H1N1, 22.2 against H3N2, and 29.1 against influenza B Victoria lineage. While the relative vaccine efficacy for influenza B had a wide confidence interval driven by the smaller number of influenza B cases accrued in that season, the point estimates for all strains were consistent. Here are the influenza B efficacy results from trials of other enhanced influenza vaccines. mRNA 1010 demonstrated a 29.1% relative vaccine efficacy against B Victoria based on 60 cases. This estimate is consistent with 25.5% for Fluzone HD based on 89 cases. Across all these trials, influenza B case numbers were limited, resulting in wider confidence intervals. Overall, mRNA 1010 influenza B efficacy estimate is consistent with the evidence used to support licensure of other enhanced influenza vaccines. As Dr. Anderson reviewed, during the 24-25 season, there was a significant H3N2 antigenic mismatch. When the efficacy analysis was restricted to the matched cases, the point estimate increased from 22.2% to 34.5%, suggesting a better match may improve efficacy. Here's the cumulative number of influenza cases over time in the study. mRNA 1010 case accrual is shown in light blue, and the standard dose influenza vaccine accrual is shown in dark blue. We see that the curves separate early after vaccination, indicating an early onset of the advantage of mRNA 1010, and the separation is maintained through the full influenza season. We also evaluated vaccine efficacy in the first versus the second half of the influenza season, and the advantage for mRNA 1010 is seen in both periods, suggesting sustained improvement over the whole season. Efficacy was consistent across age subgroups among participants 50 to 64 years, as well as in the older adults 65 and older. Even for the most vulnerable population of adults 75 years and older, the relative vaccine efficacy was 25.3%. Relative vaccine efficacy estimates were consistent with the overall outcome in participants who did and did not receive influenza vaccines in the prior season. Now, looking at more vulnerable populations, in those with one or more high-risk conditions, mRNA 1010 demonstrated a relative vaccine efficacy of 22.3%. When using the more expansive alternate definition, the RVE was 25.6%. Efficacy was consistent across frailty levels, including those meeting the criteria for frail at 30.3%. In participants with a BMI of 30 or higher, mRNA 1010 demonstrated a relative vaccine efficacy of 27.5%. We evaluated medically attended influenza in recipients of mRNA 1010 versus comparator. For those who had any healthcare encounter, mRNA 1010 demonstrated a relative vaccine efficacy of 33.7% over standard dose vaccine. For participants who required a higher level of care, either urgent care, ER, or hospitalization, events were reduced in the mRNA 1010 recipients with a relative vaccine efficacy of 47.9%. Although the case counts were not high enough to assess vaccine efficacy individually for hospitalization or ER visits, the case counts were 4 versus 8 for hospitalization and 6 versus 12 for ER visits, favoring mRNA 1010. Now, focusing on the group of participants 65 years of age and older, those with higher flu morbidity, among participants who had any healthcare encounter, mRNA 1010 demonstrated a relative vaccine efficacy of 34.1% over comparator. Relative vaccine efficacy for that higher level of care endpoint was 65.1% compared to the standard dose vaccine. The case counts were small, but the splits favored mRNA 1010 even more, 1 versus 4 for hospitalizations and 1 versus 5 for ER visits. I will now turn to immunogenicity data, starting with the study 304. These results support the efficacy findings I just presented. Immunogenicity was assessed in terms of both geometric mean titer ratios, shown on the left, and seroconversion rate differences, shown on the right, for each of the 3 influenza strains. The mRNA 1010 group had higher geometric mean titer ratios and seroconversion rate differences for all 3 vaccine strains, H1N1, H3N2, and B Victoria, with GMRs ranging from 1.59 to 1.82 compared to standard dose vaccine. The seroconversion rate differences ranged between 20 and 28% and was very comparable to the superior relative vaccine efficacy observed for mRNA 1010. We also evaluated microneutralization titers as an adjunct measure of immunogenicity. mRNA 1010 elicited higher geometric mean ratios for all 3 vaccine strains, and these results support the findings of the HAI assay. We also evaluated T-cell responses in participants receiving mRNA 1010 compared with standard dose vaccine. Shown here are the polyfunctional CD4 T-cell responses, those secreting 2 or more functional markers, at day 1 and day 29. mRNA 1010 induced a significant increase in the frequency of antigen-specific CD4 T-cells. This effect was consistent across all 3 vaccine strains. In contrast, the standard dose vaccine showed a statistically significant increase only for B Victoria. The responses at day 29 were significantly higher for mRNA 1010 compared to the comparator for all strains. Together, these data support that mRNA 1010 induces broader and more consistently polyfunctional CD4 T-cell responses compared to standard dose influenza vaccine. Now we'll discuss the immunogenicity results from Study 303. This trial was conducted in approximately 3,000 adults 65 and older. Participants were randomized 1-to-1 to receive either quadrivalent mRNA 1010 or high-dose quadrivalent influenza vaccine and were followed for 6 months. The immunogenicity here supports the accelerated approval of mRNA 1010 in adults 65 years and older. The primary objectives included evaluation of the non-inferiority of mRNA 1010 to high-dose vaccine against 4 influenza A and B strains at day 29, as well as safety and reactogenicity. A key secondary objective was to evaluate superiority of the immune response compared to high-dose vaccine. The demographics and baseline characteristics of the study population were well balanced between groups by age, sex, ethnicity, and previous vaccine status. Approximately 40% of the participants had high-risk baseline conditions by the same definition of high risk used in Study 304. Immunogenicity was assessed in terms of both geometric mean titer ratios, shown on the left, and seroconversion rate differences, shown on the right, for each of the 4 influenza strains. Superiority for mRNA 1010 was demonstrated for all strains by GMR and seroconversion rate difference against the enhanced influenza vaccine. The GMRs were 1.21 to 1.34, and the SCRs were 8.6% to 13.4%. We also conducted a post hoc analysis comparing HAI geometric mean titers between Study 303 Part C and Study 304, as the strains were similar and the results were again consistent. Antibody persistence was evaluated through 6 months in this study. Shown here are the HAI geometric mean titers, or GMTs, at month 6 for mRNA 1010 and the high-dose comparator. Titers were sustained over time and trended higher at 6 months with mRNA 1010 than with the high-dose comparator across all vaccine strains. So in summary, I've shown you data that the relative vaccine efficacy of mRNA 1010 was 26.6% higher than a licensed standard dose influenza vaccine. We met the most stringent pre-specified efficacy success criteria with a p-value of 0.0005. mRNA 1010 demonstrated consistent relative vaccine efficacy across multiple case definitions by influenza strains, by age group, by high-risk condition, and by frailty status. And the efficacy advantage was maintained throughout the whole influenza season. mRNA 1010 prevented more influenza-associated medically attended illness, requiring a higher level of care with a relative vaccine efficacy of 47.9% over the comparator. The efficacy demonstrated is consistent with the relative vaccine efficacy of enhanced influenza vaccines when compared to standard dose vaccines in other clinical trials. mRNA 1010 elicited higher immune responses compared to both standard dose and high dose comparators. We also demonstrated that HAI and microneutralization titers were strongly associated with efficacy on a group level, and CMI responses also were observed. HAI titers were sustained over time and remained higher with mRNA 1010 than with the high-dose comparator. Now I'll turn the presentation over to my colleague, Dr. Eleanor Wilson.
Thank you, Dr. Das. Good morning. I'm Eleanor Wilson, Executive Director of Clinical Development at Moderna. I'm also an infectious disease physician, and I've spent the bulk of my career in clinical research and clinical care at the University of Maryland. Today, I'll be presenting the safety data from our clinical program. In order to comprehensively evaluate safety, we assess both solicited and unsolicited adverse events in all our studies over 3 time periods. Solicited adverse local and systemic reactions are assessed over the 7 days following vaccination. All unsolicited adverse events are assessed for 28 days following vaccination. And finally, medically important events, including medically attended adverse events, serious adverse events, including death, adverse events leading to discontinuation, and adverse events of special interest, listed here, are assessed for at least 6 months following injection. First, I'll present the safety results from Study 304, the pivotal efficacy study. In this study, 6,000 participants were pre-selected to provide information on solicited adverse reactions for the first 7 days following vaccination via an electronic diary. Unsolicited adverse events were collected from all participants at every study visit and phone contacts as described. Compliance with the safety follow-up at each pre-specified time point over the 6 months was excellent, with high completion rates. Here, you can see the solicited local reactions, with mRNA 1010 shown in light blue and standard dose comparator in dark blue. While local reactions were reported more frequently in mRNA 1010 recipients, the majority of reported reactions were grade 1 and 2, mild to moderate. They were transient, and they did not cause participants to seek medical attention. A low frequency of grade 3 reactions were observed. There were no grade 4 reactions reported. Fewer and milder reactions were reported by participants aged 65 and older in both groups, but the pattern remained similar. Similarly, here are the solicited systemic reactions for the same cohort by treatment group. Systemic reactions were higher with mRNA 1010 versus the licensed standard dose influenza vaccine. Similar to the local reactions, most of these systemic reactions were grade 1 or 2 and of short duration, with a median of 2 days. A low frequency of grade 3 reactions was observed, and again, no grade 4 reactions were reported. Fewer and milder reactions were again noted in participants over the age of 65 in both groups, but the pattern was similar. All unsolicited adverse events, regardless of relationship to vaccination, were balanced between groups in the 28 days after injection. No deaths or fatal SAEs were assessed as related to vaccine. Medically attended adverse events occurred at the same rate in both groups. The frequency of unsolicited adverse events was similar through 6 months of follow-up. Importantly, there were no confirmed myocarditis, pericarditis, or myopericarditis events in either group, either within the first 28 days, the more conservative 42-day risk window, and cases were balanced through the end of study. Now, I will review the safety data from Part C of Study 303, which evaluated the safety and immunogenicity of mRNA 1010 compared to the high-dose influenza vaccine comparator in adults age 65 and older. As a reminder, this study evaluated the quadrivalent composition of both mRNA 1010 and high-dose comparator. Local reactions were reported more frequently in mRNA 1010 recipients compared to the high-dose vaccine. However, most events in both groups were grade 1 or 2, mild to moderate, self-limited, with a median duration, again, of 2 days. Systemic reactions demonstrated a similar pattern. These reactions were reported more frequently in mRNA 1010 recipients, but again, consistent with Study 304, most were grade 1 or 2, transient, self-limited, and had a median duration of 2 days. The frequency of unsolicited adverse events through day 28, again, regardless of relationship to vaccination, was similar between vaccine groups across all categories. There were no confirmed myocarditis, pericarditis, or myopericarditis events in either group, either within the first 28 days or through 6 months of follow-up. It's important to note there was one fatal event in an mRNA 1010 recipient, which was determined by the study investigator to be unrelated to vaccination. Finally, in discussion with the FDA, we conducted an integrated summary of safety across the entire Phase 3 program, which included all adults aged 50 and older from all 4 Phase 3 studies. This pooled analysis included nearly 72,000 participants, 36,000 mRNA 1010 recipients, and a similar number of participants who received either standard or high-dose comparator vaccines. All available safety data follow-up was included through 6 months or 1 year, depending on the length of follow-up in each study. The analysis evaluated deaths, serious adverse events, and adverse events of special interest. No safety concerns were identified in this integrated safety analysis of more than 71,000 participants. Through day 28, the frequency of serious adverse events, fatal adverse events, and adverse events of special interest was low and similar across vaccine groups. Results were also similar through 6 months of follow-up. So in summary, mRNA 1010 has demonstrated an acceptable safety profile compared with standard dose and high-dose influenza vaccines in adults aged 50 years of age and older. Solicited local and systemic adverse reactions were reported more frequently with mRNA 1010 than with comparator vaccines. Most of these events were mild, grade 1 or 2 in severity, and resolved within 2 days without medical attention. The most common reactions were injection site pain, fatigue, and headache. Medically attended adverse events, serious adverse events, fatal events, and adverse events of special interest occurred at similar rates between mRNA 1010 and standard dose or high dose vaccine recipients. Overall, the safety profile was acceptable in both adults aged 50 to 64 and adults 65 years of age and older. I'll now return the presentation to Dr. Das.
Thank you, Dr. Wilson. I'll now summarize the benefit-risk profile of mRNA 1010. As discussed today, influenza remains a serious and potentially life-threatening illness, and currently licensed influenza vaccines are an important foundation of public health. However, vaccine effectiveness remains variable across seasons and can be affected by strain mismatch as a result of manufacturing timelines, antigenic drift, and egg adaptation. mRNA 1010 was designed to address these challenges. It directly encodes the selected hemagglutinin antigens, avoids egg-based manufacturing, and leverages the flexibility of the mRNA platform. Together, these features support the potential for improved protection against influenza and its significant morbidity and mortality. As a reminder, we are seeking approval of mRNA 1010 for adults 50 years of age and older. It's administered as a single intramuscular dose in a prefilled syringe, and the application includes 2 regulatory pathways: traditional approval for adults 50 to 64 years of age, and accelerated approval for adults 65 years of age and older. Overall, the benefit risk supports approval in all adults 50 years of age and older. Across the clinical program, mRNA 1010 demonstrated a favorable benefit risk profile in adults 50 and older. In Study 304, mRNA 1010 demonstrated superior relative vaccine efficacy compared to standard dose vaccine, with consistent protection across influenza strains, age groups, and high-risk groups. These clinical efficacy results were supported by higher immune responses compared to standard vaccine and superior immune responses compared to high-dose vaccine in adults 65 years of age and older. mRNA 1010 also had an acceptable safety profile. Local and systemic reactions were more frequent than the comparator, but they were generally transient and resolved without medical attention. No safety concerns were identified. Taken together, these data support the potential for mRNA 1010 to reduce the influenza burden beyond currently licensed vaccines. For adults 50 to 64, the efficacy advantage demonstrated in Study 304 should translate into fewer influenza cases, fewer medically attended illnesses, and fewer hospitalizations. For adults 65 and older, we believe mRNA 1010 has the potential to provide protection that is comparable to currently available enhanced vaccines. In addition, the superior immune responses observed versus high-dose vaccine suggest the possibility of additional clinical benefit, and this will be tested directly in our planned randomized real-world effectiveness study. Beyond the clinical findings, mRNA 1010 also offers potential platform advantages. One is the ability to move strain selection closer to the influenza season, reducing the time available for circulating viruses to drift away from the vaccine strain. Modeling studies suggest that this could improve strain matching in a meaningful proportion of seasons and potentially prevent thousands of influenza-related hospitalizations during mismatch years. A second potential advantage comes from the non-egg-based manufacturing approach. Multiple studies have shown higher effectiveness for cell-based compared to egg-based influenza vaccines. Because mRNA 1010 is also a non-egg-based platform, it has the potential to provide similar benefits by avoiding egg-adaptive changes that can contribute to strain mismatch. While the magnitude of these benefits will vary from season to season, together, they illustrate how both the clinical performance of mRNA 1010 and the underlying advantages of the mRNA platform could contribute to improved protection against influenza. We are committed to generating the direct comparative effectiveness data for mRNA 1010 versus licensed enhanced influenza vaccines in adults 65 years of age and older. To do this, we are planning a large randomized pragmatic real-world effectiveness study in an integrated health system. The study is expected to enroll 400,000 adults, 65 years of age and older per season, randomized to receive either mRNA 1010 or enhanced influenza vaccines. Both groups will have the same follow-up for laboratory confirmation of influenza illness. The primary endpoint will be vaccine effectiveness against laboratory-confirmed medically attended influenza. Key secondary endpoints will include effectiveness against urgent care, ED visits, and hospitalizations. It will also assess effectiveness by influenza type. We are finalizing the study design with the FDA to confirm and expand our understanding of the benefit of mRNA 1010 compared to enhanced influenza vaccines. And we intend to begin components of the study prior to licensure. The evidence presented today also supports a positive benefit-risk profile across both regulatory pathways. For adults 50 to 64 years of age, the need for improved influenza prevention remains, particularly among individuals with high-risk conditions. In this group, mRNA 1010 demonstrated statistically superior efficacy compared with standard dose vaccine. The efficacy findings were supported by higher immune responses, and the safety profile was acceptable with increased reactogenicity that was generally short-lived. These data support traditional approval in adults 50 to 64 years of age. For adults 65 years and older, where the burden of influenza is highest, need remains despite the availability of enhanced vaccines. In this age group, mRNA 1010 demonstrated efficacy compared to standard dose vaccine in a range consistent with licensed enhanced influenza vaccines. mRNA 1010 also demonstrated statistically superior immune responses compared with high-dose vaccines with an acceptable safety profile. Together with the potential for improved strain match through the mRNA platform applicable for both age groups, as well as Moderna's commitment to confirm the benefit of mRNA 1010 in this population, the totality of data support accelerated approval in adults 65 years of age and older. Moderna is committed to continuing to generate clinical and safety data for mRNA 1010. We have already conducted studies in adults 18 to 49 years of age evaluating immune response and safety. We also have studies planned to fulfill our pediatric commitment with the FDA, and we will initiate a dose-ranging study in children and adolescents 9 to 17 years of age shortly. Following licensure, routine pharmacovigilance activities will be conducted to assess vaccine safety in real-world use. These data will be shared with the FDA and other agencies as they become available. In summary, the data presented today support approval of mRNA 1010 for the prevention of influenza A and B in adults 50 years of age and older. On behalf of my colleagues at Moderna, thank you for the opportunity to present today. We also would like to thank our study investigators, site personnel, Data Safety Monitoring Board, and most importantly, the study participants. We will be happy to address any questions.
Thank you very much, Dr. Das. We're going to have 10 minutes now for questions from the committee. Dr. Durbin.
Thank you. Thank you. I just have two quick questions. One, in terms of the mRNA platform, was the LNP used for mRNA 1010 the same LNP that's been used in other Moderna COVID vaccine or Moderna mRNA vaccines, which will give us sort of a broader safety base as well? And secondly, were CD8 T cell responses measured at all in any of the studies? I know it wasn't presented, but I'm just curious. Thank you.
Yes. So for the LNPs, this is indeed the same as the COVID, as both of our COVID vaccines, as well as the RSV vaccines. As a reminder, over 6 billion people have received the COVID vaccines over the last 5 years. In terms of the CD8 T cells, we did measure CD8 T cells. There was a CD8 T cell response. As expected, though, the magnitude was lower than the CD4 T cell response.
Thank you.
Dr. Omer.
Yeah, I have a couple of questions. Did you do the stratified analysis by location specifically? What were the results, given the sort of the differences in comparative vaccines and sort of epidemiology? So I'm specifically interested in US data. You should be able to do some subgroup analysis with reasonable sample sizes there. Then a second question is, I couldn't, neither from the briefing document or the presentation, I couldn't see, sort of decipher what were the differences specifically in grade 3 reactions between the two groups. And then the last one is, I hope there is consideration after the deliberations and recommendations of this committee, there is a renewed consideration of existence of clinical equipoise in your post-marketing randomization study, because considerations of clinical equipoise are seminal to being able to randomize irrespective of the stage of the study. But that's I think that may require a longer conversation, but the first two are quick questions.
Okay, yes, absolutely. So we did do our analysis by geography. So here's the geographic analysis. And for North America, which was very similar to the US analysis, it was quite consistent with the advantage of mRNA 1010 over the standard dose comparator. We, in your question, I'll also answer there was a comparator, trivalent versus quadrivalent comparator difference in some of the European geographies. We did analyze by trivalent versus quadrivalent comparator, and there was no difference in the efficacy advantage in the RVE point estimate. I will quickly show the grade 3 responses, and then if there's more detailed questions, I'll bring up my colleague, Dr. Wilson. Oops, sorry, I think I put the wrong one up. Okay. Sorry, here we go. And so this is the difference in the grade 3 responses. So this is for local reactions. So for the individual local reactions, the grade 3 responses, the grade 3 reactions, ranged from 0.3% to 0.9% for the mRNA 1010. And then I'll show you the systemic as well. And here are the systemic grade 3 reactions. And they ranged from 0.2 to 3.2% for mRNA 1010. Now, in terms of the clinical equipoise question, we are planning a randomized study in a large health system, and so could you tell me a little bit more about the question of clinical equipoise?
So the question is that if there is a recommendation, without preempting the recommendations vis-a-vis folks older than 65 years, this could change the clinical equipoise around the question of whether it is appropriate to continue to randomize. I'm not sort of rendering a summary ethical verdict, in this Q&A session, but I'm just raising the question that, going forward, I hope there are considerations of actual clinical equipoise whether there is significant uncertainty to justify the act of randomization, even if it's a large health system activity, other than the sort of the other option could be a more standard observational study. But that may be, admittedly, a longer conversation.
Yes. So please keep the answer brief. We only have under 10 minutes now.
Yes, indeed. And so, yes. And so the randomization is intended to be, with enhanced vaccine, I think underlying is that the clinical benefit that we're seeing in our clinical development program is in line with enhanced vaccines. And so so provided that the recommendation is there, the randomization can proceed on that front. And so we are working very diligently with our partners along with the FDA to ensure the success of this study.
Thank you. Dr. Gans.
So I know that we only have a short time in this, so I'm hoping that
we're going to continue
from Moderna will continue the conversation.
Yeah, so we're going to continue the conversation this afternoon. So Don't feel anybody's going to be cut off. Dr. Gans, go ahead.
So the very relevant safety question that I wanted to raise at this point, there's a lot of other things to consider, but I didn't see what was defined in your evaluation of any adverse events. I'm particularly interested in neurologic outcomes. There was a call out for all of the cardiac related ones, but obviously for previous influenza, we have other signals that would be of interest with the messenger RNA, such as Guillain-Barré and other encephalopathies.
Yeah, indeed. And so we did evaluate that quite carefully, and I'll bring up Dr. Wilson for that.
Thank you, and thank you for the question. Yes, in our predefined adverse events of special interest, we evaluated inflammatory neurologic events, and those included Bell's palsy, Guillain-Barré, and Guillain-Barré acute demyelinating disease and seizures were included in our definition. When we evaluated those events in our overall safety analysis, both by study and in the integrated summary of safety, there was no safety signal for any inflammatory neurologic events. Events were balanced in both arms.
Thank you. Dr. El-Sali.
Thank you, Arnold. Question pertains to mRNA 1010. What is the difference between this mRNA 1010 and the one that was evaluated in the preceding season on which data were published in 2025?
Indeed. Thank you. And that's an important question to answer. So the mRNA 1010 formulation was optimized here. There were point mutations in the non-antigenic regions introduced for the B strain in order to increase antigen expression. And the effect, it indeed had the desired effect. As you'll recall, in the prior studies, it was a bit equivocal whether we were achieving superior B immunogenicity compared to the comparators. And in this study, both 303C as well as 304, we noticed the superior B immunogenicity now, and also the B efficacy.
But the A did not change between both.
The A's did not change.
Okay.
Thank you. Dr. Munoz.
Thank you, Dr. Monto. Just one quick clarification question and then a second follow-up. The clarification is for your immunogenicity endpoints and VE endpoints for the 65 and over, it's clearly stated that they were done at 28 days, so within a month after vaccination. That was not clear to me for the standard dose study. Could you comment on when those VE assessments were done?
So the efficacy assessment was over the full influenza season, and so the median was 6 months, but it was when they got vaccinated up till the end, April 30th, so up to 8 months, and that persistent efficacy was seen throughout the whole season.
Okay, thank you. That's very helpful because you did show that slide with the rapid within a month VE, and then it seemed like it plateaued. But this is VE overall, not a particular time point.
Yes, indeed. And then when we split it, first half, second half, we also saw that the advantage of mRNA 1010, the superiority of efficacy was also maintained.
Perfect. And the other quick comment relates to also the composition. So you have a very nice reactogenicity in terms of low side effects, low reactogenicity from this vaccine. And, people would want to compare this with COVID vaccines. And so with your optimization that you just described regarding the immunogenicity, can you comment on all the potential changes that also could result in lower reactogenicity?
So, we didn't study it head to head. And so the side-by-side reactogenicity, it's not a randomized, so it's not totally a fair comparison. When we line the numbers up to mNEX spike, they are, I would say they're comparable.
And you have a different dosage as well.
Yes. This is a 50 microgram dose, and the mNEX spike is that optimized NTD RBD. Yep.
Okay, thank you. Dr. Perlman.
Yeah, just a question on the follow-up study that you're going to do. We had discussed earlier about the issue of frail individuals and immunocompromised. Is there going to be any special efforts to particularly incorporate them into the follow-up study?
So, we can certainly consider enrichment strategies, but the follow-up study is going to be large, and there will be certainly no exclusion for immunocompromise. And the intent, and we believe we will be, we will have enough people to do the sub-analyses here as well.
Okay, thank you.
And this will be similar to the recently published study about high dose versus adjuvant, correct?
Yes, absolutely. Absolutely.
Okay, thank you very much. Next, we go over to the FDA presentation, and it's going to be led by Dr. Raval and Dr. Brennan.
Thank you, Dr. Monto, and good morning. My name is Godi Raval, and myself and Dr. Timothy Brennan are medical officers here in the Office of Vaccines at the FDA, and we will be presenting the FDA review of the efficacy, immunogenicity, and safety of Influenza, or mRNA 1010. Next slide. This is the outline of our presentation, and I will present the introduction, the overview of clinical studies, and our review of the key efficacy and immunogenicity data. And then I'll turn the presentation over to Dr. Brennan, who will discuss our review of the safety data, risk mitigation plans, and finally, a summary and voting questions for VRBPAC. Next slide. Influenza, or mRNA 1010, is an mRNA-based seasonal trivalent influenza vaccine. It's composed of 3 mRNAs, each containing 12.5 micrograms, encoding the hemagglutinin, or HA glycoproteins of influenza subtypes A, H1N1, A, H3N2, and type B Victoria lineage, encapsulated in lipid nanoparticles. It is administered as a single dose of 0.375 mL administered intramuscularly, and the applicant's proposed indication is for the active immunization for the prevention of influenza virus subtypes A and type B represented in the vaccine. The applicant's proposed approval pathway includes traditional approval for adults 50 through 64 years of age and accelerated approval for adults 65 years of age and older. Next slide. First, I'll review the clinical studies that contributed to the BLA submission and specifically to the FDA review presented today. Next slide. The applicant's clinical development program included 4 phase 3 studies that are summarized here. The 2 studies that I will discuss more in depth today are emphasized in the green boxes, and they are first study P304 and second study P303 Part C. Study P304 was the pivotal phase 3 relative efficacy study conducted in adults 50 years of age and older, which evaluated the final trivalent or TIV formulation of mRNA 1010 that is intended for licensure. It was evaluated against a standard dose influenza vaccine, and it contributes the relative efficacy data to support traditional approval in adults 50 through 64 years of age and supportive efficacy data for the accelerated approval in adults 65 years of age and older. Study P303 Part C was conducted in adults 65 years of age and older, and it contributes the immunogenicity data supporting accelerated approval in this older age group. This study evaluated the quadrivalent or QIV formulation of mRNA 1010 against a high-dose influenza vaccine that is preferentially recommended in adults 65 years of age and older. The QIV formulation of mRNA 1010, other than the inclusion of the additional mRNA encoding for the HA glycoprotein for the B Yamagata strain, was otherwise identical to the trivalent formulation used in study P304. Studies P303 Parts A and B and studies P301 and P302 shown here contributed only to the safety review through the integrated summary of safety. Studies P301 and P302 use the original formulation of mRNA 1010 with influenza B antigens manufactured differently from those in the formulation intended for licensure, while studies P304 and P303 use the final formulation intended for licensure. Next slide. I'll now review the key efficacy data from study P304. Next slide. The relative efficacy data that I will review today from study P304 forms the basis for the applicant's proposed traditional approval pathway in adults 50 through 64 years of age, and it provides supportive relative efficacy data for the proposed accelerated approval pathway in adults 65 years of age and older. Study P304 was a phase 3, randomized, observer-blind, active controlled trial evaluating the relative efficacy, safety, and immunogenicity of mRNA 1010 TIV. Over 40,000 participants 50 years of age and older were randomized 1-to-1 to either receive mRNA 1010 or a standard dose influenza vaccine comparator, which was either Fluarix TIV or Fluarix QIV. Fluarix TIV was the preferred standard dose comparator and was used in North America. Fluarix QIV was used where TIV was not available in Europe and East Asia. The study was conducted during the 2004 to 2005 Northern Hemisphere influenza season at 301 sites in 11 countries. The primary analysis of relative vaccine efficacy for this study was conducted on April 30th, 2025, which was considered to be the end of influenza season. This was the initial pre-specified interim analysis for the study, but as 968 cases had been accrued by then, which exceeded the target of 836 cases, the study did not continue into a second influenza season, and this analysis used the full one-sided alpha of 2.5%. The primary endpoint of the study was the first episode of RT-PCR confirmed protocol-defined influenza-like illness with onset 14 days or after vaccination through the end of influenza season, caused by any influenza A or B strain with RT-PCR confirmation within 7 days of symptom onset. The statistical success criteria for the primary endpoint included sequential testing for pre-specified non-inferiority, superiority, and super-superiority thresholds, which were the lower limit of the 95% confidence interval of the RVE being greater than minus 10% for non-inferiority, greater than 0% for superiority, and greater than 9.1% for super-superiority. The thresholds and terminology for superiority and super-superiority were defined by the applicant. Next slide. Study P304 study design is further described here in a timeline of study activities experienced by each participant. Prior to vaccination on day 1, there was a pre-vaccination blood draw for all participants for baseline immunogenicity responses, and a second blood draw for immunogenicity responses occurred on day 29 post-vaccination. In terms of safety monitoring, a subset of approximately 6,000 participants, referred to as a reactogenicity subset, was monitored for solicited local and systemic adverse reactions. All participants were monitored through 28 days post-vaccination for unsolicited adverse events. And all participants were monitored for 6 months after vaccination for serious adverse events, medically attended adverse events, and adverse events of special interest, and through study duration, which was approximately 6 to 8 months, for serious adverse events that were considered related to vaccination. Active surveillance was conducted for all participants for protocol-defined respiratory illness symptoms throughout the study. The data cutoff for the primary efficacy analysis was April 30th, 2025, based on the end of influenza season. All participants had a final or end of study visit, which may have occurred at either the end of influenza season visit or at the day 181 or 6-month post-vaccination visit, whichever came later. The final study visit for the final participant occurred on August 21st, 2025, and this was the date through which all safety analyses were conducted. Next slide. The demographics and baseline characteristics of participants in study P304 are summarized here and were balanced across the mRNA 1010 and standard dose comparator groups. Across both groups, the median age of participants was 64 years of age, with a range of 50 to 97 years. 52% of participants were 50 through 64 years of age, 48% were 65 years of age and older, and 12% were 75 years of age and older. The majority, 70% of participants, were from sites in North America. Nearly half, 47% of participants had received the seasonal influenza vaccine in the previous season. And the majority, 57%, had at least one high-risk condition at baseline. The most common high-risk condition was an elevated BMI greater than 30. Among the participants 65 years of age and older, the majority, 73%, were considered fit and not vulnerable or frail based on the Edmonton Frail Scale. And to address the prior question asked by Dr. Durbin, although participants considered vulnerable or frail were enrolled in the study, fewer than 1% were considered moderately or severely frail with a frailty score of 10 or higher. In addition, the study did not collect data on nursing home or assisted living residents. Therefore, the generalizability of study findings to very frail older individuals may be limited. Next slide. Shown here is the analysis of the primary hypothesis-tested endpoint of relative vaccine efficacy of mRNA 1010 compared with the standard dose comparator against RT-PCR confirmed protocol-defined ILI caused by any influenza A or B strain in participants 50 years of age or older. This study evaluated relative vaccine efficacy, which measures how much mRNA 1010 reduced the risk of the specific outcome when compared to another U.S. licensed standard dose vaccine. It does not evaluate absolute efficacy, which would require comparison to a placebo. Relative vaccine efficacy can therefore be thought of as the additional or added benefit of mRNA 1010 beyond the protection provided by the comparator vaccine. There were 411 cases of protocol-defined ILI confirmed by RT-PCR testing in the mRNA 1010 group, compared with 557 in the standard dose comparator group. This resulted in a relative vaccine efficacy of 26.6%, or put another way, a close to 27% reduced risk of PCR-confirmed protocol-defined ILI as compared with the standard dose comparator. Based on the lower limit of the 95% confidence interval, which was 16.7, all three of the applicants' pre-specified success criteria were met, including non-inferiority, superiority, and super-superiority. This analysis was based on a median efficacy follow-up time of 181 days, or approximately 6 months. Next slide. Study P304 also evaluated relative vaccine efficacy against RT-PCR confirmed protocol-defined ILI caused by any influenza A or B strain by age subgroup as a pre-specified descriptive analysis that is shown here. The RVE remains fairly consistent across subgroups, ranging from about 25% to 28%. In the 50 to less than 65-year-old age group, the 95% confidence interval excludes 0, and although not hypothesis tested, suggests superiority against the standard dose comparator. In adults 65 years and age older, age and older specifically, the RVE point estimate of 27.4% remained robust and consistent with that seen in the overall study population and younger age cohort. The demonstrated lower bound of the 95% confidence interval of 12.1 also suggests superiority over the standard dose comparator. However, it does warrant consideration that the comparator is a standard dose influenza vaccine, which is not a preferentially recommended vaccine in this age group, and an efficacy comparison against a preferentially recommended influenza vaccine is not available. Further subdividing the participants who are 65 years of age and older, those who are 65 to less than 75 years of age continue to have an RVE consistent with the full study population and a confidence interval that excludes 0 and suggests superiority. Participants 75 years of age and older made up the smallest age subgroup with just over 4,500 participants across groups. The smaller number of participants likely resulted in a wide 95% confidence interval that crossed 0. Therefore, although the RVE remains directionally consistent, the confidence interval does raise uncertainty about the RVE in this age group. Next slide. Study P304 also evaluated the relative vaccine efficacy against RT-PCR confirmed protocol-defined ILI by influenza strain type in a pre-specified descriptive analysis. The RVE point estimates remain robust and range from 22 to 29% across either influenza A subtype and individually for influenza A, H1N1, influenza A, H3N2, and for B, Victoria. Influenza A strains predominated in the study, accounting for 94% of all influenza cases accrued and driving the overall RVE estimate. RVE was slightly lower for influenza A, H3N2, though the 95% confidence interval still excludes 0. Influenza A, H3N2 showed the highest antigenic mismatch among strains, with 44 to 48% of cases antigenically mismatched. So this may be a contributor to the slightly lower efficacy observed. Data on antigenic mismatch across strains will be discussed further in the next slide. In the case of B Victoria, there were very few cases accrued, with 60 cases across both groups in the season. Although the RVE point estimate remained consistent at 29.1, the 95% confidence interval was very wide and crossed 0, likely due to the small number of cases, which limits interpretation of this RVE. Next slide. Shown here is an analysis of RVE against RT-PCR confirmed protocol-defined ILI with antigenic match, which was a descriptive pre-specified analysis. Overall, antigenic match between the circulating and vaccine strains varied by strain. For influenza A, H1N1, about 98% of cases were antigenically matched. For B Victoria, about 94% of cases were matched. An antigenic match was lowest for influenza A, H3N2, with 52 to 56% of cases antigenically matched. For influenza A, H3N2, RVE against antigenically matched strains was higher at 30.5% compared with the RVE against all strains, which was 22.2%, which may suggest that efficacy of mRNA 1010 against cases of H3N2 may be higher in seasons with greater antigenic concordance. For the other influenza strains, the RVE point estimates were relatively consistent, comparing all strains and antigenically matched strains. For influenza B Victoria, there were only 34 cases with antigenic typing results across the group that contributed to the analysis. So again, very low case accrual resulted likely in a wide 95% confidence interval that crossed 0, which limits interpretation of this RVE. Next slide. Study P304 also evaluated the relative vaccine efficacy against a different influenza-like illness case definition, modified CDC-defined ILI, which was evaluated using hypothesis testing with an endpoint of RT-PCR confirmed modified CDC-defined ILI. Modified CDC-defined ILI is a more stringent case definition compared with the study's protocol-defined ILI, as it requires a temperature above 37.2 degrees Celsius or 99 degrees Fahrenheit and cough and/or sore throat. With 223 cases in the mRNA 1010 group and 290 cases in the standard dose comparator group, the RVE against modified CDC-defined ILI was 23.5%, with a lower limit of the 95% confidence interval of 9.0. Therefore, using the same pre-specified hypothesis criteria that were used for the primary endpoint, both non-inferiority and superiority were met, but the threshold of superiority was not met against the standard dose comparator. Pre-specified descriptive subgroup analyses based on age groups was also conducted for this endpoint, and the RVE point estimates remain robust and directionally consistent, with the lower bound of the 95% confidence intervals remaining above 0 in both adults 50 through 64 years of age and those 65 years of age and older. In particular, analysis of participants 65 years of age and older still demonstrates a robust RVE of 26.7% with a lower bound of 4.4 when using this more stringent case definition. Next slide. Finally, the study evaluated RVE against health outcomes associated with RT-PCR confirmed protocol-defined ILI, including healthcare encounters beginning 30 days after respiratory symptom onset, as a descriptive pre-specified analysis. For any healthcare encounter associated with protocol-defined ILI, which included hospitalizations, ER visits, urgent care visits, and outpatient clinic visits, the RVE was 33.7%. RVE specifically for cases who were seeking a higher level of care that included hospitalizations, ER visits, and urgent care visits was 47.9%. When looking at urgent care visits and outpatient visits individually, while the RVE was directionally consistent, the 95% confidence interval crossed 0. While this study was not powered to evaluate healthcare outcomes associated with ILI, these exploratory results suggest that mRNA 1010 may offer greater efficacy than the standard dose comparator in preventing more severe influenza-associated illness. This effect appeared most pronounced in participants 65 years of age and older, in whom relative vaccine efficacy for healthcare encounters seeking a higher level of care was 65.1%, with a 95% confidence interval of 17.4 and 85.2, with a case split of 7 cases in the mRNA 1010 group and 20 in the standard dose comparator group. Next slide. The previous slide, thank you. That concludes our review of the efficacy data from study P304, and now I'll move on to the immunogenicity data. Next slide. The immunogenicity data to be reviewed today forms the basis of the applicant's proposed accelerated approval pathway in adults 65 years of age and older, and comes from study P303, Part C. Study P303 Part C was a phase 3, multi-center, randomized, stratified, observer-blind, active-controlled study evaluating the safety and immunogenicity of the quadrivalent formulation of mRNA 1010. Over 3,000 participants 65 years of age and older were randomized to either receive mRNA 1010 QIV or Fluzone high-dose QIV, which is a preferentially recommended influenza vaccine in adults 65 years of age and older. The study enrolled healthy adults and adults with stable chronic diseases, and participants were stratified by previous season influenza vaccination. The study was conducted during the Northern Hemisphere 2003 to 2004 influenza season at 96 sites in the United States. HAI antibody responses based on the hemagglutinin inhibition, or HAI assay, were measured at baseline pre-vaccination, day 29, and post-vaccination, and then at day 181 or end of study in a subset of participants. The primary immunogenicity analysis was based on HAI antibody responses at day 29 post-vaccination, with the primary endpoints being HAI geometric mean titer, or GMT, and seroconversion rate, or SCR, at day 29 for all 4 vaccine-matched influenza strains. The study's statistical success criteria were tested sequentially, with superiority evaluated after non-inferiority had been demonstrated for all 8 co-primary endpoints. For non-inferiority, the pre-specified criterion for success was a lower limit of the 95% confidence interval greater than 0.667 for each strain. And for seroconversion difference, a lower limit of greater than -10% for each strain. For superiority for the GMT ratio, the criterion was a lower limit of the 97.5% confidence interval being greater than 1.0 for each strain, and for SCR difference, the lower limit of greater than 0% for each strain. Next slide. The demographic and baseline characteristics of participants in study P303 Part C are shown here and were balanced across the mRNA 1010 QIV and Fluzone high-dose QIV groups. The median age of participants was 70 years, with a range of 64 to 93 years. The majority of participants, 78%, were 65 to less than 75 years of age, and 22% were 75 years of age and older. Just over half, 53% of participants, had received a seasonal influenza vaccine in the prior season. High-risk factors were assessed in participants post-hoc based on medical history, and 39% of participants had at least one high-risk factor, most commonly diabetes mellitus. Next slide. Study P303 Part C's primary endpoint of GMTs as measured by HAI for vaccine-matched influenza strains at day 29 is shown here. The far right column includes first the GMT ratio, followed by the 95% confidence interval, which was used for the non-inferiority evaluation, and then the 97.5% confidence interval, which was used for the superiority evaluation. Based on the lower limit of the confidence interval for the GMT ratio, both non-inferiority and superiority of mRNA 1010 were met against Fluzone high dose for all 4 strains. Next slide. The primary endpoint of seroconversion differences as measured by HAI for vaccine-matched influenza strains at day 29 are shown here. SCR is defined as the proportion of participants with either a baseline HAI titer prior to vaccination of less than 1 to 10 and a post-vaccination titer greater than or equal to 1 to 40, or a baseline titer of greater than or equal to 1 to 10 and a minimum 4-fold rise in titer post-vaccination. The primary endpoint was the difference in the seroconversion rate between the 2 groups. The far right column shows first the difference in seroconversion rate, followed by the 95% confidence interval that was used for the non-inferiority threshold, and then the 97.5% confidence interval that was used for the superiority threshold. Based on the lower limit of the confidence interval for the difference in seroconversion rate, both non-inferiority and superiority of mRNA 1010 were met against Fluzone high dose for all 4 strains. Next slide. The study also included a descriptive analysis of GMTs at day 181 or end of study, which are shown here. GMTs remained numerically higher in the mRNA 1010 group compared with the Fluzone high dose group for all 4 strains, though the confidence intervals overlapped for 3 strains, and only influenza A, H3N2 demonstrated non-overlapping confidence intervals. Next slide. A descriptive analysis of the seroconversion differences at day 181 or end of study are shown here. The percentage of participants with seroconversion at day 181 was higher in the mRNA 1010 group compared with the Fluzone high dose group across all 4 strains, but with overlapping confidence intervals for all strains except for influenza A, H1N1. Together, the GMT and seroconversion data suggest persistence of immune response at day 181 after vaccination with mRNA 1010 QIV. Next slide. So the immunogenicity data I've just reviewed from study P303 Part C supporting accelerated approval in adults 65 years of age and older uses the quadrivalent formulation of mRNA 1010, not the trivalent formulation proposed for licensure. Therefore, the applicant conducted a post hoc descriptive analysis of mRNA 1010 TIV and QIV formulations to justify using data from QIV to support licensure of TIV. That data is shown here, and it compares the day 29 HAI GMTs induced by mRNA 1010 TIV from study P304 to the day 29 HAI GMTs induced by mRNA 1010 QIV from study P303 Part C for each of the 3 shared influenza strains restricted to participants 65 years of age and older. The model did account for differences in baseline demographic factors between the 2 groups, including age, race, sex, ethnicity, baseline BMI group, previous influenza vaccine status, and baseline high-risk status. Although the analysis was not hypothesis tested, based on the GMT ratios and the lower limit of the 95% confidence interval, non-inferiority of the TIV formulation would have been demonstrated for all 3 shared strains using a threshold of the lower limit of the 95% confidence interval greater than 0.667. For influenza A, H1N1, the GMT ratio of TIV to QIV approaches 1, indicating no meaningful difference between the TIV and QIV formulation. For influenza A H3N2 and influenza B Victoria, the TIV formulation elicited higher antibody responses than QIV, with the GMT point estimates and the lower limits of the 95% confidence interval both greater than 1.0. Lower antibody responses from the quadrivalent formulation may suggest that inclusion of B Yamagata may have reduced immune responses to influenza A, H3N2, and influenza B Victoria strains, which favors mRNA 1010 TIV in producing higher immune responses. There are several important limitations to this analysis. The 2 studies were conducted in different influenza seasons with different strain selection, and there is a potential for residual confounding despite adjustment for baseline characteristics. Recognizing these limitations, FDA's preliminary assessment is that because the data supports non-inferiority of TIV to QIV across the 3 shared strains, with any interference from B Yamagata favoring TIV, the immunogenicity data from mRNA 1010 QIV, which demonstrated superiority against a high-dose comparator, may be used to support the effectiveness of mRNA 1010 TIV in adults 65 years of age and older. Next slide. So that concludes our review of the immunogenicity data, and now I'll turn the presentation over to Dr. Brennan to discuss the safety data. Thank you.
Good morning. My name is Timothy Brennan, and I will be discussing our assessment of the safety of mRNA 1010, as well as a pharmacovigilance plan and post-marketing required study. Finally, I'll summarize our overall assessment of mRNA 1010 and reiterate the VRBPAC voting questions. This slide here summarizes the sources of safety data used in our assessment of the safety of mRNA 1010. The two primary sources for the safety evaluation are Study P304 in adults 50 years of age and older, and Study P303 Part C in adults 65 years of age and older, with each of these studies having approximately 6 months of follow-up. As the applicant had conducted 4 phase 3 clinical studies, we recommended also conducting an integrated safety analysis, or ISS, pulling safety data from all 4 clinical studies in adults 50 years of age and older. The ISS population comprised approximately 72,000 participants, with approximately 36,000 participants in both the mRNA 1010 and the standard dose or high dose comparator groups, with a median follow-up of 198 days in both groups. The pooled mRNA 1010 group includes participants who received trivalent or TIV and quadrivalent or QIV formulations across studies, including the previous formulation of the vaccine with the B strain formulated differently. The pooled comparator group includes standard dose TIV and QIV formulations and high dose QIV licensed influenza vaccines. Next slide. Thank you. This table summarizes key findings that characterize the overall safety profile of mRNA 1010. As I will show in more detail in the next few slides, solicited local and systemic adverse reactions were more frequent in the mRNA 1010 group compared to the comparator group. However, rates of unsolicited adverse events through 28 days, serious adverse events, deaths, and adverse events of special interest through a median follow-up of 198 days were overall balanced across the mRNA 1010 and comparator groups. The pooled ISS analysis across all 4 phase 3 studies did not identify adverse event patterns definitively indicative of a safety concern for mRNA 1010. Next slide. Shown here are the frequencies of solicited local reactions following a dose of mRNA 1010 or the standard dose comparator vaccine in adults 50 years of age and older from Study P304. As mentioned in the prior summary slide, the overall rate of solicited local adverse reactions was higher in the mRNA 1010 group at 67.5%, compared with the standard dose comparator group at 32.1%. The majority of solicited local adverse reactions were mild to moderate, though grade 3 solicited local adverse reactions were more frequently reported in the mRNA 1010 group. There were no grade 4 solicited local adverse reactions in either group. Next slide. Shown here are the frequencies of solicited systemic adverse reactions following a dose of mRNA 1010 or the standard dose comparator vaccine in adults 50 years of age and older from Study P304. Similar to solicited local adverse reactions, the overall rate of solicited systemic adverse reactions was higher in the mRNA 1010 group at 58%, compared with the standard dose comparator group at 32.4%. The majority of solicited systemic adverse reactions were mild to moderate. The rates of grade 3 solicited systemic reactions were higher in the mRNA 1010 group compared to the standard dose comparator group. There were no grade 4 solicited systemic adverse reactions in either group. In the mRNA 1010 group, the majority of solicited local and systemic adverse reactions occurred within 2 days following vaccination and persisted for a median of 2 days. Next slide. Shown here are the frequencies of solicited local reactions following a dose of mRNA 1010 or the high dose comparator vaccine in adults 65 years of age and older from Study P303 Part C. Similar to the overall pattern seen in Study P304, the overall rate of solicited local adverse reactions was higher in the mRNA 1010 group at 66.1%, compared with the high dose comparator group at 38.9%. The majority of the solicited local adverse reactions were mild to moderate, though rates of grade 3 solicited local adverse reactions were higher in the mRNA 1010 group compared to the high dose comparator group. There were no grade 4 solicited local adverse reactions in either group. In the mRNA 1010 group, the majority of solicited local adverse reactions occurred within 2 days of vaccination and persisted for a median of 2 days. Next slide. Shown here are the frequencies of solicited systemic reactions following a dose of mRNA 1010 or the high-dose comparator vaccine in adults 65 years of age and older from study P303 Part C. Again, similar to what was observed in study P304, the overall rate of solicited systemic adverse reactions was higher in the mRNA 1010 group at 61.3%, compared with the high-dose comparator group at 32.9%. The majority of the reported solicited systemic adverse reactions were mild to moderate, though grade 3 solicited systemic adverse reactions were more frequently reported in the mRNA 1010 group. And there were 2 grade 4 solicited systemic adverse reactions of fever reported in the mRNA 1010 group. Finally, in the mRNA 1010 group, the majority of solicited systemic adverse reactions did occur within 2 days following a vaccination and persisted for a median of 1 day. Next slide. Over the full study period, with a median follow-up of 198 days, serious adverse events, or SAEs, were reported in 3.1% versus 2.9% of participants in the mRNA 1010 group and comparator vaccine groups, respectively. Within 28 days post-vaccination, SAEs occurred in 0.5% of participants in each group. 3 preferred terms, or PTs, were numerically higher in the mRNA 1010 group and had risk differences that excluded 0. These were death unspecified with 23 versus 9, urinary tract infection or UTI with 25 versus 12, and anemia with 9 versus 2. All of these being reported in less than 0.1% of participants in either group. Of note, there were also 3 preferred terms that were numerically higher in the standard dose or high dose comparator groups and had risk differences that excluded 0. These included chronic kidney disease at 0 versus 4, urinary retention, 0 versus 4, and hypertensive emergency at 0 versus 4. Over the full study period, deaths due to any cause were balanced and reported in 0.3% of participants in each group. Within 28 days of vaccination, deaths were few and comparable across groups at 13 versus 4. Next slide. As mentioned in the prior slide, a numerical imbalance was identified in the preferred term death unspecified cause, with 23 cases in the mRNA 1010 group versus 9 in the comparator group. When pooling related death categories such as sudden death, sudden cardiac death, and unspecified fatal events, the combined analysis identified a total of 29 mRNA 1010 recipients versus 12 comparator recipients. Despite the numerical imbalance, there are several characteristics that suggest that this imbalance is unlikely to represent a causal relationship with vaccination, including the median time to onset of 131 days in the mRNA 1010 group with no temporal clustering. The balance observed in deaths within 28 days following vaccination at 3 versus 2. The presence of significant cardiovascular and metabolic comorbidities in most participants. The balanced rate of preferred terms that are frequently associated with sudden death, such as myocardial infarction, 7 versus 7, cardiac arrest, 6 versus 6, cardiovascular accident, 4 versus 5, and sepsis-related deaths at 5 versus 6. The absence of investigator-assessed relatedness of the deaths to vaccination in all but one case. This was a case of a 76-year-old female with a prior myocardial infarction and coronary artery bypass grafting, atrial fibrillation, type 2 diabetes, who died suddenly on study day 2, with the investigator assessing the death as related due to the proximity of the death to vaccination. Our assessment in this case is that the participant's significant cardiac history represents a more likely etiology. Taken together, our assessment is that the totality of the available evidence does not suggest that the numerical imbalance in death unspecified is likely to represent a vaccine safety signal for mRNA 1010. Next slide. An expanded analysis across related anemia preferred terms, including anemia of chronic disease, blood loss anemia, hyperchromic anemia, iron deficiency anemia, and normocytic anemia, identified 14 mRNA 1010 recipients and 8 comparator recipients with anemia SAEs. Most of these events occurred more than 90 days post-injection, with a median time of onset of 134 days in the mRNA 1010 group with no temporal clustering. A review of medically attended adverse events, or MAAEs, of anemia in study P304 identified comparable rates between the groups, with 31 mRNA 1010 versus 30 in comparator recipients. Similarly, an expanded analysis across related urinary tract infection, or UTI, preferred terms, urinary tract infection bacterial, Escherichia urinary tract infection, cystitis, kidney infection, pyelonephritis, acute pyelonephritis, and urosepsis identified 38 mRNA 1010 recipients and 22 comparator recipients with UTI SAEs. Most of these events also occurred more than 90 days post-injection, with a median time to onset of 135 days in the mRNA 1010 group, again with no temporal clustering. A review of medically attended adverse events of UTI in study P304 identified comparable rates between the groups, with 153 mRNA 1010 and 158 recipients in the comparator group. None of the anemia or UTI SAEs were assessed as vaccine-related by the investigator. Taken together, our assessment is that the late onset, the absence of temporal clustering, presence of recognized risk factors in the majority of affected participants, and the balanced MAAE rates in study P304 suggest that the numerical imbalance in anemia and UTI SAEs is unlikely to reflect a causal association with mRNA 1010. Next slide. Throughout the full study period, adverse events of special interest, or AESIs, were reported in 36, or 0.1%, versus 37, also 0.1%, of participants in the mRNA 1010 and comparator groups, respectively. There were no cardiac AESIs, such as myocarditis, pericarditis, or myopericarditis, or cases of Guillain-Barré syndrome reported in the 42-day risk window. There was one case of Bell's palsy reported in each group within the 42-day risk window. Next slide. And now I will discuss our risk mitigation assessment, which includes the planned pharmacovigilance and post-marketing required study. Based on the totality of the safety data, there are no important identified or potential risks or missing information requiring additional pharmacovigilance beyond routine surveillance. Therefore, the applicant plans to conduct routine post-marketing pharmacovigilance surveillance activities, including the submission of periodic safety reports to monitor for and assess any emerging risks associated with the vaccine. Additionally, the applicant will provide aggregate safety assessments of the following AESIs in their periodic safety reports for the first 3 years post-approval: thrombocytopenia, Guillain-Barré syndrome, acute disseminated encephalomyelitis, Bell's palsy, seizures, anaphylaxis, myocarditis, pericarditis, and myopericarditis. The details of the pharmacovigilance plan remain subject to discussion between the FDA and the applicant. Next slide. Under the accelerated approval regulations, if Influenza is approved for use in individuals 65 years of age and older, a confirmatory study will be required to verify and describe the clinical benefit of Influenza in individuals 65 years of age and older. The applicant has proposed to conduct a phase 4 cluster randomized, active controlled, pragmatic study to evaluate the relative vaccine effectiveness of Influenza compared with an agreed upon CDC preferentially recommended vaccine. Calls for vaccine clinics to alternate weekly between Influenza and the comparator vaccine throughout the influenza season. Approximately 800,000 or 400,000 participants per season, adults 65 years of age and older, will be randomized 1-to-1 to either Influenza or the comparator vaccine for 2 full seasons. Next slide. And now I will conclude with an overall summary of our assessment of mRNA 1010, as well as our voting questions for the VRBPAC members. Next slide. In summary, mRNA 1010 demonstrated superior relative vaccine efficacy versus standard dose comparator in adults 50 years of age and older, and superior immunogenicity versus a CDC preferentially recommended comparator in adults 65 years of age and older. The reactogenicity profile, while elevated relative to the comparators, was predominantly mild to moderate and resolved within 2 days. Overall rates of SAEs, deaths, and AESIs were balanced between mRNA 1010 and comparator groups. Data limitations include the single season of follow-up, the absence of data in immunocompromised individuals, limited data in very frail older individuals, and no concomitant vaccine data. These limitations, however, are not unique to this application, and some will be addressed with ongoing safety surveillance through post-marketing pharmacovigilance and verification of clinical benefit in adults 65 years of age and older through a cluster randomized phase 4 confirmatory study. Next slide. Excuse me. This final slide contains the voting questions for the VRBPAC members. Number 1, do the benefits of Influenza outweigh its risks for the prevention of influenza disease in adults 50 through 64 years of age? Vote yes, no, or abstain. And number 2, do the benefits of Influenza outweigh its risks for the prevention of disease in adults 65 years of age and older? Yes, no, or abstain. Finally, Dr. Raval and I would like to take a moment to thank our many colleagues within OVRR and across CBER who have contributed to an incredible amount of time and effort to ensure a thorough review of this application prior to our presentation to the VRBPAC members. This truly is a team effort, and we have an amazing team of dedicated reviewers across all disciplines who have helped make this presentation possible. Thank you very much.
Thank you both. We're going to have just a few minutes of urgent questions because we have a hard stop because of the open public hearing. So we'll have a few questions and continue in the afternoon. Yes, Dr. Gans.
I'll pose this now and happy to have it discussed later. So thank you for that presentation. On slides 31 and 32, you walked us through some of the data on the difference in ratios of some of the unexpected adverse events. And I'm wondering if you have more information. I realize you're probably pulling these off of this, but since this must have had a DSMB and we should know more about those events, and they were considered by the quote investigator not to be related. So for instance, was there breakthrough disease in any of these? So they had actually influenza, or were there other infections or other reasons for these being reported, but then, subsequently not being considered vaccine related? So I think that would be important data to help us understand the nuance, particularly of the deaths due to any cause, the unexpected.
The unexpected deaths, yes. We do have a backup slide.
Okay.
60. Yeah, we do have a backup slide that addresses that. Okay, great. So this slide in particular is focusing within 28 days of vaccination. So you can see that even amongst the deaths unspecified, there are very few deaths within that 28-day period, and it's relatively balanced with 3 in the mRNA 1010 group versus 2 in the comparator group. In each one of these cases, the medical history included significant comorbidities that could be associated with sudden death, sudden cardiac death, as well as some concomitant medications that could have contributed as well. But I would say that the biggest issue that we faced when evaluating these is the lack of data, the lack of autopsy data, the lack of additional medical information. Some of these patients had withdrawn consent, for example, and there was no additional information from the hospital. Some of these patients were found deceased at home, so there was no additional information beyond that, and no autopsy performed. And so I would say that, perhaps maybe after the break, Moderna could also address their considerations, their thoughts about these deaths unspecified.
Thank you, and thank you all. We're going to break for lunch until 1 o'clock Eastern Time.
Dr. Monto, we're live.
Afternoon. Yeah, I'm ready. We're going to begin the afternoon session with the open public hearing. We're slated to have an hour available, but it may take less time to go through it. So I want to alert everybody that we will be continuing with the rest of the agenda when the open public hearing session is completed. And I would like to welcome all of you to the open public hearing session. Please note that both the Food and Drug Administration and public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee, the FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the public hearing speaker, at the beginning of your written or oral statements to advise the committee of any financial relationship that you may have with the sponsor, the product, and if known, its direct competitor. For example, the financial information may include the sponsor's payment of expenses in conjunction with your participation in this meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address the issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. Over to you, Dr. Cecily.
Thank you, Dr. Monto. The FDA and this committee place great importance in the open public hearing process. The insights and comments provided can help the agency and this committee in their consideration of the issues before them. One of our goals for today is for the open public hearing to be conducted in a fair and open way where every participant is listened to carefully and treated with dignity, courtesy, and respect. Therefore, please only speak when recognized by myself or the chairperson. We will now begin with OPH speaker number 1. Please unmute and introduce yourself.
Hello, I am Don Ford, and I have no financial relationships with anyone FDA approves anything for, but I wouldn't mind that. Anyways, whenever you're ready. So I wrote this slide deck using comparative data originally between what we saw as the best flu vaccine compared to the comparators and reverse. So I'm going to go off the data that they showed us today, which showed an improved response. So ignore some of my commentary about it not being as good, though I do feel it should have more attention. Next slide. I walked in saying no, but it just feels not ready. We watched an hour of comparative data to the standard dose that I feel like could have been an email. We're looking at a vaccine for approval for 55 plus. I think entertaining the idea of looking at anything except for the ACIP recommended vaccines is a giant waste of time. I think it's making the product look better than it is, and that it might not be harmful on paper, but it will be harmful socially because the people who jump on these things are going to jump all over this. It's going to be a total mess. So I really wanted to say that. I also want to say that we talked a lot about mRNA doing quick updates, and Moderna specifically talked at a COVID meeting, specifically said that that was dependent upon the government paying for the updates. If the government wasn't paying for the updates, because it's the waste issue. And so when we talk about this as a possibility, at best you're looking at choosing the variant later, which is a benefit, but there's no clear mechanism inside the FDA to accomplish this. This is something that we've been through with trying to move the COVID vaccines up because COVID is not a seasonal thing that appears in the winter. It appears more in the summer. We really need vaccines on a different timetable. And right now there's no mechanism to make that happen. And it's a big problem. One of the regulators referred to Moderna's study as theirs. That made me question the safety data and whether or not it was being handled objectively, which makes me think that this is something that should be reevaluated over time. The A is too high. Level 3A shouldn't be allowed at all. I do think that all of these things are going to damage uptake at a time where flu vaccine uptake is at an all-time low. And the way that this has been handled online since then is an absolute mess. And I'll go to the next slide. The product, if it is approved, it should be approved with clear advertising limits, because right now we have people all over the internet comparing it to the standard flu dose, and the public does not understand what the standard flu dose is. They don't understand that it's a sort of outdated metric that we need updated, which I advocated for at the last meeting. So if you guys do choose to approve this, please do it where they are not allowed to do it in comparison to the standard flu dose. Is going to be a enormous mess. Next slide. On that note, please update the standard flu shot metric to our friends at CBER. It's confusing. Like I said, there's no middle ground on this. We have to meet the public where they are. It's unreasonable to expect millions of people to keep up with us. And this allows influencers and all sorts of people who are not directly affiliated with the products to say all kinds of things that aren't necessarily true, and that can often lead people into harm. And again, that will damage uptake more. Next slide. You guys mentioned this already. I said the mRNA vaccines need to have a clear improvement. There is a move. It's a very vocal move to remove our very effective vaccines with what could potentially be less effective mRNA vaccines. And we have the similar problem with the DTaP vaccine. The DTaP vaccine wasn't studied long enough to determine if it had a long-lived plasma cell response that lasted more than a few years. That was a problem. I would hate to see that repeated with our extremely effective vaccines at a time when we're seeing immune suppression from COVID infections. Next slide. I'm always going to advocate for protein-based vaccines. I have no relationship with any protein-based vaccine manufacturers. At this point, it's kind of silly that I don't, but I don't, and it's ridiculous that we're kind of going the wrong directions with vaccines, where we're comparing, where they keep saying, oh, this is as good as the previously approved vaccine, but that was approved then, and we're here now. Now we should see an improvement. So if we're not seeing an improvement, we can't compare the old status quo to the new status quo. That is actually going backwards, kind of like inflation. If your income doesn't increase based on inflation, you're actually making less money every year. And the same thing is true in the quality of our vaccines. Next slide. Non-related influenza, final point to our friends at CBER. I'm going to bring this up in every meeting forever until it's done. Nuvaxovid for pediatric populations. It's an expansion of the same vaccine. I know it's not influenza, but they submitted their documentation, and this really needs to happen. So please, 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 let's make this happen. Parents need it bad. I'm a parent. I need it badly. Next slide. That's it. I'm done. Thank you for your time. Thank you as always, everyone. You're doing a fantastic job. Great presentations, everyone, all around.
Thank you, sir. Open public hearing speaker number 2 will not be participating, and we will pick up with open public hearing speaker number 3. Please unmute yourself and introduce.
Yes. Good afternoon. My name is Wellington Sun, and I was the director of CBER's Vaccine Division from 2008 to 2018. But I was also previously employed by Moderna in 2018 to 2020. However, my comments today are completely my own and without influence or compensation for my comments from any other party. The purpose of my comments today is to really provide a brief historical perspective to FDA licensure of seasonal influenza vaccines within the past 2 decades, with emphasis on the regulatory criteria used to establish effectiveness and the approval pathway used. Slide 1. Okay, thank you. Of the 3 possible pathways for demonstrating efficacy, the traditional or full on the left, and accelerated are used the most for vaccines. CBER puts a great deal of weight on the disease, clinical disease endpoint as evidence of efficacy. Pre-specified success criteria are set high enough that a single successful randomized controlled trial provides enough confidence that the vaccine is effective. Such RCT is truly the gold standard. Accelerated approval uses a surrogate marker, in the case of flu, the hemoglobin inhibition titer, to show that the vaccine is likely effective. Note that CBER has always considered HI titers as likely to predict clinical benefit, and not a correlate of protection that implies more certainty of effectiveness. Hence, there is a requirement for post-licensure RCT to verify clinical benefit for accelerated but not for the additional approvals. Slide 2. Over the last 2 decades, there has been a dramatic increase in available licensed flu vaccines, spurred on by a critical shortage in the 2004-2005 season, during which the U.S. was down to a single manufacturer. The 2009 H1N1 pandemic added greater urgency for additional manufacturing technology than the conventional egg-based. Note the wide use of both approval pathways, and sometimes the rationale can seem complex. Slide 3, please. Here are some highlights that occurred at CBER during this expansion of licensed flu vaccines. There was a flurry of accelerated approval from 2005 to 2009. Many of those were vaccines already marketed overseas in Europe and Australia. There are 3 first-in-class vaccines using new manufacturing technology: CellVax with cell-based, block, recombinant protein, adjuvant. Successful clinical disease endpoint RCTs always resulted in a conditional approval, either for the initial licensure or as the required post-licensure RCT to verify clinical benefit after accelerated approval. The design of clinical disease endpoint RCTs allowed use of either placebo or licensed non-flu vaccine comparator for evaluating absolute efficacy, or a licensed flu vaccine comparator for evaluating relative efficacy. And recognizing that the unpredictability of the dominant serotype for any particular season, CBER allowed use of clinical flu due to all subtypes combined as RCT primary endpoints in all of these clinical endpoint studies. CBER used HI titer antibody also for traditional approval for expansion of age indications, but only after previous successful clinical efficacy of randomized controlled trial had been done. So this process has been referred to as immunobridging. And CBER exercised discretion in allowing approvals based on RCTs and immunogenicity endpoint versus each-day, but not all met. Finally, I'd like to end by emphasizing the unique role of this committee in CBER to serve as gatekeeper for allowing only safe, pure, and potent vaccines into the market. But also, there's an unspoken and unappreciated responsibility to enable a diversified and robust ecosystem of flu vaccines to protect against seasonal and future influenza. Thank you.
Thank you, sir. Open public hearing speaker number 4 will also not be participating in today's open public hearing, so we'll have open public hearing speaker number 5. Please unmute yourself and introduce.
Yes. Hi, good afternoon. Can I be heard okay?
Yes.
Excellent. Thank you. I'm Dr. Michael Abrams, Senior Health Researcher at Public Citizen, a nonprofit consumer advocacy organization with over 1 million members. We have no financial conflicts of interest regarding today's topic. The clinical data regarding mRNA 1010, of course, brand name Influenza, and the FDA's analysis of that data support the vaccine's traditional approval for individuals aged 50 to 64. The clinical data further support the limited accelerated approval of the vaccine for those aged 65 and older. Safety and effectiveness both seem reasonable in the 50 to 64-year-old population, demonstrating that the mRNA vaccination can prevent serious flu-related morbidity, including medically attended illness, as well as are better than existing flu vaccines with rare risk of serious adverse effects. For those aged 65 and older, the data, of course, are less certain, as we've heard, largely because the comparison treatment did not use CDC-recommended high-dose adjuvanted or recombinant vaccine formulations for this older population. However, blood markers of the immune response, in fact, 2 forms, are reassuring that the mRNA vaccine is plausibly protective. Additionally, a large post-marketing study is planned to confirm the vaccine's safety and effectiveness over 2 seasons through 2029. Safety and effectiveness confirmation will be especially important in people older than 65 years of age and those who are immunocompromised or otherwise have pre-existing conditions, as we saw regarding the imbalances of unspecified death in particular. It is essential that the post-marketing study be completed on the agreed-to schedule and that the findings are promptly published and reported to the public. Public Citizen notes that the FDA's VRBPAC, this committee, has now met twice within a 1-month period. We urge the FDA to continue to frequently convene this committee, as well as many other expert advisory committees that the agency has since January of 2025 insufficiently utilized. We also applaud the FDA for the scientific consideration of an important public health vaccine that uses mRNA technology. Public Citizen and many others outside the agency remain deeply concerned that the FDA will reach decisions about important drugs, biologics, and medical devices without public scrutiny and sufficient input from outside experts, such as people on this panel today. We hope that today's advisory committee on this flu vaccine will be a harbinger for the agency's approach moving forward. Thank you very much.
Thank you for your comments. Dr. Monto, that was our last speaker. You may now close the open public hearing.
Thank you. As predicted, we're finished with the open public hearing, so we are moving on to additional Q&A for CDC, FDA sponsors, and other presenters. Raise your hands, as I'm sure you will. Dr. Durbin.
Thank you. This is a question for FDA. Given that one of the purported advantages to this technology is rapid change of strain, should it be necessary, my question to FDA is, how would that be implemented if, for instance, there was a new strain emerged that differed from the recommended strains at the usual VRBPAC meeting? How would a manufacturer work with FDA, or how would FDA allow for different strains than was recommended by VRBPAC? Is there a process in place?
On? Good. Thank you, Dr. Durbin, for that question. Clearly, that's an important question. Clearly, if we want to take advantage of more rapid manufacturing technologies, that needs to be addressed. I would say that we're under discussion. Is that on? Oh. Now can you hear me?
Yes, thank you.
Okay. Sorry if you didn't. You didn't catch my answer, I take it.
I did. I think I did. It's under discussion.
Okay. Yes, that was the answer. Thank you.
And I might point out it's rather complicated because it's a question of it is a multifaceted issue. Dr. Gans.
Well, I do want to echo what Dr. Durbin said, because I think that's actually important. And the conversation continues on that vein to consider, as we talked about at the last VRBPAC meeting for COVID, whether or not there also could be additional strains added if there was another signal that we saw that we wanted to do. So I think the under discussion needs to be a little broader than just, changing the exact H1N1 or H3B that are already in there. So from 3 to 4 again, or 4 to 3, or whatever we feel the data is saying to us. My real question really comes about another topic that I think we have to start to think about. And I really appreciate Moderna giving us a broader immunologic look. So we had both humoral, although I did have a question about actually bringing the raw data of the humoral immune responses forward for us to look at. There was a full difference looking at this compared to the comparator. But the reason why I bring it up is because there is also, we have had to choose, because of the efficacy of this vaccine that we currently have available, which is showing and sort of clinically is demonstrating lower T-cell responses. That brings up the question of persistence immunity, boosted immunity over multiple seasons, and potentially the need to not have to choose every year if there is a lasting component of that. So I think what I'm trying to say is we really need to dig into two things. We really need correlates of protection. So there was some allusion that, that we're sort of looking at that. There was some data that was presented that greater than 1 to 40 on the assays that we use is protective, but we really actually don't understand that. So I think what we would ask from any manufacturing partners would be, when you have breakthrough disease, we should be looking at least the closest humoral immunity, or if they come, since these are medically attended, if they could actually have blood drawn so that we could actually understand what their closest humoral immune response was, to say like that was not protective against however we define breakthrough disease. Did they have lower HI, for instance? And we should be looking at their post responses. So in that conversation, I think we really have to understand that. I would also urge, even though you've looked at one season, which I think the data, given the number of cases there were, is able to support the data you brought forward for the non-inferiority and superiority. However, we really need to understand, again, the persistence. So we do need season 2. So I'm wondering what we're doing with this cohort and looking through that, particularly, and this might be a biased population who gets revaccinated, but we sort of need those correlates to understand how good messenger RNA may be at establishing T-cell immunity, which would support a better memory response. So that's my first question. I'll go back in the queue for anything else.
Okay, I'm sure you will.
So we can, if we can. Yeah.
So is this, Haley, is this directed to Moderna?
Yeah, it's Moderna, and also to the FDA, sort of for the broader consideration of some of the conversation.
So Moderna first.
Yeah, so thank you for that question. And we certainly agree that that it is quite important to understand the immunology of flu as well as the protective markers. And so I'll show you the CD4 immunogenicity, which we were very privileged to do in the phase 3 study. And we saw very good responses that were higher than the standard dose comparator. You also asked for the raw titers and not just the GMFRs, and the best display I have right now is the reverse cumulative frequency distribution curves. And here, so this is, I'll orient you a little bit. So red now is mRNA 1010, and blue is the active comparator. And And on the bottom are the proportions of people achieving those HAI titers, so 1 to 10, 1 to 20, 1 to 40, and so forth. And then I have it for H1N1 all the way on the left, H3N2 in the middle, and B Victoria on the right. And as you can see that the red curves, which are the Moderna, the mRNA 1010 distributions, are shifted to the right. So they are persistently higher. And then on the histograms on the bottom, you do see the shift in all of those histograms to the right as well, that those immune responses are persistently higher as well. In terms of the correlates of protection analysis, and I think you're absolutely right. The totally ideal thing to do is to get the blood work at the time of case. We were not able to do that in the study, which it is quite operationally challenging to do that because people get care at different places. We did do the day 29 collections and then went back on everybody in the trial and then went back and got the cases and non-cases. Those analyses are still ongoing, but in general, they show that the stronger HAI responses that we are seeing from mRNA 1010 is correlated at least directionally with protection, and the same story is seen from the microneutralization and the CD4 T cells. We also had some more detailed immunology data published just this week from Dr. Alabetti's group on lymph node and germinal center responses. And I'll bring up Dr. Carol Henry from our research group to detail those.
Thank you, Dr. Das. So I'm Dr. Carol Henry. I'm in infectious disease research at Moderna. I'm an immunologist by training. As Dr. Das pointed out, we did an observational study that was conducted at Washington University by Dr. Ali Alibadi and colleagues, where we collected for some individuals that were vaccinated with mRNA 1010 compared to a standard dose Fluarix vaccine, where we looked more deeply at B cell responses. So we looked at memory B cell responses post-vaccination. Also, fine needle aspirations were collected to look at the germinal center responses of these individuals post-vaccination. And what we could see in these individuals is that in the serological titers, they had higher magnitude of responses. So the people that received mRNA 1010 compared to Fluarix. But also they had higher frequencies of memory B cells when they received mRNA 1010, and very interestingly, they had sustained germinal center reaction that were not found with the standard dose flu vaccine. And these, sustained germinal center reaction, as you might know, we found that memory B cells re-enter those germinal centers in mRNA 1010 vaccinees, and that correlated with serologically with higher breadth of responses in mRNA 1010 vaccinees in both H1N1 and H3N2. So a broad panel of viruses were tested. So I think this is a good example of, sustained immune responses and how B cells diversify in the germinal center and basically better adapt the immune responses with mRNA vaccines.
Any further comments from FDA or CDC? Oh, you've got another speaker. Please go ahead. Moderna.
Oh no, I'm sorry. I was just coming back to moderate.
Okay. Okay. Any further questions from or comments from FDA or CDC? Because I know CDC is trying to do some of these studies as well.
So I'm not sure we have our CDC.
Do we have? Yeah, that was my next question.
I'm not sure we have the correct CDC,
We'll
but I can speak from the FDA perspective. And as it relates to the broader question of correlates, sponsor will know that from at least our office, we have and we continue to plan to encourage evaluations of correlates of risk and to collect the more comprehensive data that's needed to establish a correlate of protection. And even more helpful is to establish a threshold of protection that can serve as a clinically validated surrogate of endpoint. So the bottom line is, Dr. Gans, I think we well receive your message on the importance of establishing correlates of risk, even more so correlates of protection, and at the end of the day, a threshold of protection that could be seen as a clinically validated surrogate endpoint.
Thank you, Dr. Omer.
Thank you. This question could either be answered by our FDA colleagues or our Moderna colleagues. So I want to confirm that in the planned randomized trial, there is adequate power to detect a difference in mortality of a similar magnitude as the numerical difference in the between the deaths of unspecified cause. I did a quick back of the envelope calculations, and it does seem to be the case, but I didn't have the precise details of the pooled analysis in which the difference was detected. So I would like to confirm that there's sufficient statistical power of that study, which is as I understand, is to be conducted over 2 flu seasons. Second, that this is indeed an endpoint because the background documentation shared with us and presented does not specifically say that. So I just want to confirm that it is an a priori endpoint to explore a difference between the two groups.
Dr. Das?
Yeah, I can start, and then I'll bring up Dr. Anderson, who leads our epidemiology group, and will be running the study along with our partners. And so, bringing back the study design, oh, yes, thank you. Yes, and so we do have so it will be large, conducted over 1 to 2 seasons. The powering is based on the medically attended influenza, but that said, it is quite a large study, and we will be capturing a multitude of influenza outcomes, including hospitalizations and deaths, and in terms of the all-cause mortality, I will let Dr. Anderson give details. I don't believe all-cause mortality is an endpoint in this study, but we do routine safety surveillance, and so it would be captured in that portion of the study as well. Dr. Anderson, is there anything else you would add to that? No? Okay. All right. Yep. So we will capture it as part of the safety surveillance.
Thanks for providing the response.
Okay. Dr. El-Sali.
Thank you, Arnold. I have a question pertaining to the 2-year duration of the clinical trial 304. Dr. Das indicated that it was planned for 2 years, actually, and then that is no longer the case. So was that decision the FDA's, Moderna's, or agreed upon by both parties?
Yeah, so the study was actually intended to collect 836 cases. And so we initially planned that it would take us 2 influenza seasons to collect that many cases. And so because of the high severity of the 24-25 influenza season, we actually captured 968 cases. And so when we went to the DSMB for what was to be an interim analysis, all of the hypotheses were met, and that was indeed a final analysis. So we will be, of course, in the PMR, we will be conducting through additional influenza seasons or maybe more. But that was the reason that study 304 was not continued for a second season.
Thank you for that clarification. So it was event-driven. You met the event and exceeded it by some. So that's statistically understandable. However, if there's anything that is predictable, it's the unpredictability of influenza. In the season preceding the one in which the study was performed, the mRNA 1010 was evaluated, and there was no the immunogenicity, which was demonstrated to be superior for flu A. In this study, the 304, was not demonstrated, especially against H1N1, where the ratio was 1.0. So, I mean, I say this, and I don't know that there's a solution, except that we did miss an opportunity to really understand the performance of the product pre-licensure by virtue of not going through with the planned study over 2 years.
No, I certainly take your point. And I think if I can have CO13 up, it is quite, the influenza seasons are quite variable from one to another. And I think yes, influenza seasons are quite variable, and your point is well taken. I do want to say that there was the B optimizations, but you're asking about A. There were further optimizations to the UTR, which was kind of seen for our Spikevax versus mNEX spike. So it is an overall optimized vaccine candidate. The GMRs for immunogenicity are a bit better, are better for A also with this vaccine. That said, we are quite committed to the PMR study and do feel like we will get a lot of information.
Okay, thank you. I have a question on the reactor. I can come back or
no, go ahead. We don't have that many on the list.
Okay, thank you. So I definitely reassuring to see the safety data, and especially when it comes to the adverse events of special interest, where there was no difference in a large number of patients between active comparator and mRNA 1010. However, it remains important to highlight that there were 10 times as likely grade 3 local reacto, any local reacto in the 50 to 64-year-old, and 5 times as likely grade 3 systemic reacto in the same age group. And that difference remained also in the older age group. So while this is not a safety concern, this pertains to an important acceptability concern, especially in light of alternatives, and especially in light of it being an annual product. So this is this has to be messaged and highlighted in our approach if we are to increase the vaccine coverage in our population, which unfortunately has been dwindling in the last couple of years.
And I would like to add to that question about how you are going to message, because at least anecdotally, the same people who seem to have systemic reactions to the COVID vaccine seem to have them again. And we really need to be able to message why people should get this vaccine if at some point it gets a preferential recommendation.
Yeah, thank you, Arnold. I did my own little scribbling from the data presented and the files that were shared. And we have grade 3, that's incapacitating, 2 to 4% fatigue, myalgia, and arthralgia. So we as clinicians fight the notion, oh, you gave me the flu, buy your flu shot all the time. This is not going to help. So we really need to be careful and proactive about this.
Yeah, I do think both of your points are very well taken, and we are committed to very clear and open communication about the vaccine. It will happen for providers through the label, but we are also engaging multiple through the usual channels. We are also engaging vaccinators to make sure they are educated on the data. Absolutely, the grade 3s are higher, but the safety profile is very reassuring. And it is short-lived that even for the grade 3s, the duration is 1 to 2 days. But your point about very clear communication is certainly well taken. And it is also a communication that needs to be made on the totality of data, the risk versus benefit. The benefits are also in terms of the higher medical care avoidance. There's benefits and risks, and we do believe the benefits outweigh the risks, but the communication is quite important.
Okay, thank you.
Right, Dr. Perlman.
Yeah, so I have a question about, there are two related questions, one about pediatrics and one about mucosal immunity. So my read of the mRNA vaccines, mostly from SARS-CoV-2, is that they don't induce very good mucosal immunity unless somebody's been previously infected with the virus, so that there's some pre-existing mucosal immunity, then you get a boost. So do you have any information about mucosal immunity after the mRNA vaccine? And second part of that is in terms of children under the age of 7, I think it was. I saw that you weren't asking for approval. Is that part of the reason that you're not, or is there other reasons?
So for the mucosal immunity, we don't have any data for the flu vaccine mucosal immunity right now, but I think that is an important thing to study in a respiratory vaccine. So point very well taken. Our step down of age, we have a full pediatric plan agreed upon with FDA. And so this is the first part of it. The 9 to 17 study is going to start in the fall, but we will, our commitment is from 6 months up, as is typical for other flu vaccines. So we will be looking at the full spectrum of pediatric age.
Okay, thank you.
Dr. Monto, you're muted.
See what happens when you have a cough? You mute yourself and then forget. Dr. Gans.
Thank you. So I wanted to continue on the safety sort of discussion that we're having. And I really appreciated Hannah's nod to what are we going to do with increased reactogenicity. So a couple of questions. Well, one concept related to that wasn't brought up. I mean, and you alluded to this, I think, Dr. Da, but we need to really understand that if you have a good immune response to the vaccine, so therefore you're better protected, you may actually have a reaction. And those sort of combination, because when I tell people that, they're very surprised to hear that. So we clearly have not gotten the message out that actually is showing that your immune system is responding to that. So anyway, if we're going to message, but more my question, so that's a comment that I really think we have to get out there. But I wondered, when you were doing your dose finding in your phase 1/2, did you look at anything lower than 12.5 to see both the immunogenicity and reactogenicity in any populations? I see you nodding your head. So that's one question related to safety. I think I'm going to ask my questions now, if that's okay, since I would then move again.
No more than 3.
Yeah, no, they're all related to safety. But anyway, I also think that another concept that continues not to come forward to the public of understanding the totality of protection from vaccines. So if we could show in some of our safety data also long-term, particularly cardiovascular. So there's a lot of comments about influenza disease causing these other events that happen that people don't understand are necessarily linked, not only within 7 days, they're protracted. So I think we have to look more broadly to say, even if you get influenza breakthrough disease with these vaccines, because we know severity of disease is lower, then your chance of getting X, Y, and Z are, I'm presuming, would be lower. So I think we have to get that conversation into our data so people understand they're not only protecting themselves from maybe getting COVID and severe COVID, but also it's, other potential sequelae that come later. And so that's my second.
That's number one. Okay.
I did want to ask a little bit about your recruitment. So if we're having trouble messaging, this gets back to messaging. I'm wondering how your recruitment, you recruited a lot into these studies. You have a very ambitious recruitment for the next one. I'm wondering how those conversations are going, because that would also help physicians and everybody else if this were to come to licensure, how we should talk about that. So it would be helpful to understand what you're hearing out there.
Yeah, and so I wrote down the question, so please let me tell me if I'm missing something. So for dose finding, we did a whole range of doses, and we've really found that 12.5 micrograms per dose was the best balance of reactogenicity and immunogenicity. That said, we are dose ranging again for younger populations, and so the 9 to 17-year-old study that we're about to start will dose range again. And so we will kind of customize the dose finding to age as we can. The point about the totality of protection and looking at these other outcomes, all-cause mortality, all-cause hospitalization, all-cause cardiovascular mortality is really quite important. And I think, it relies on our data, but kind of everybody else's data as well. And, we know there's great data for flu vaccines preventing other adverse outcomes like Dr. Anderson mentioned. And so we do hope in this large study with the 400,000 per season to start to get at that impact. I think we're starting to get at that impact with our COVID vaccine data. I think we just showed some, are about to publish some data about cardiovascular protection from COVID vaccines. In terms of recruitment for these studies, we are working with our partner who has a lot of experience in these large real-world effectiveness studies, and they have demonstrated. We've done some other work with them, but they have demonstrated the ability to recruit like this. These numbers are very large, but they are very skilled at doing this. So we do feel confident in the ability to recruit this study.
If I may just add another question. I know reactogenicity is dose dependent, but mRNA vaccines seem to bring a bit more reactogenicity with equivalent doses. Have you looked at exactly what in the enveloping or whatever is related to this? And we might say the same for cardiomyopathies and.
Yeah, no, and we have thought about this. We have thought about this a lot, and some of it we've thought about what is the contribution of the lipid nanoparticle, which is kind of almost like the contribution of the lipid nanoparticle, as well as the antigen specifically.
Almost an adjuvant.
Yeah. Yes, and then versus the antigen, and I think in this study we looked very carefully for myocarditis and pericarditis, and we have a huge database, 71,000 people, and we didn't see it. I think a lot of the non-mRNA COVID vaccines also had a myocarditis, pericarditis signal, so maybe that is an antigen related piece, although I don't know that anybody has shown that definitively, but we are looking at this quite carefully, and when we make the dose decision, we really try to make the dose decision as carefully as possible to balance reactogenicity and antigenicity. And we will also do the same as we go down in age to make sure that these vaccines are tolerable for younger age groups as well.
Thank you. Dr. Patel.
Thank you. Thanks, Dr. Monto. So I'll just first start by thanking everyone for their really comprehensive presentations today. Very helpful to see both FDA and Moderna present on this material. I want to echo earlier comments about strain selection, where looking forward to FDA sharing with us how they plan to operationalize some of these processes. I know there is a trade-off between making sure the strain is as close to possible as a match to what we expect will be in the upcoming season, but also making sure the production timelines for the non-mRNA vaccines are reasonable so that we don't have a shortage in the fall. My question, though, is for Moderna and about the comparator arm in the 65 plus group. So I believe 70% of the sites were in North America, and given that the enhanced flu vaccines were preferentially recommended in June of 2022 in the United States, and in Canada, I believe it was July of 2024. I was curious if at all it would be possible, or have you looked at doing exploratory analyses of relative vaccine effectiveness comparing the mRNA 1010 to the enhanced flu vaccines? I know that this is the plan for your phase 4, but curious if there's any data that you were able to share related to RVE with the enhanced vaccines as a comparator.
No, we don't have the data. We don't have the data now. In our immunogenicity study, the 303C, we did not collect cases because it was just so small. Relatively small. We have done some modeling work based on the predicted efficacy benefits based on the immunogenicity, but that is a bit speculative, so I don't want to go there at this point. But that said, we are committed to doing the phase 4 post-marketing study.
Okay, thanks, Dr. Doss. And Dr. Monto, just to let you know, Dr. Grosskopf is available if when you're ready for the CDC questions.
Why don't we get Dr. Grosskopf, since she's been waiting around, to tell us about the year in question, 2004, 2005. It was kind of mixed, as I recall.
It was kind of mixed. Thank you, Dr. Monto. So looking back at the 2024-25 VE, given the context of today's discussion, I'm going to focus on adults overall 18 and up and adults 65 and
also H3N2 diversity.
I did not realize that was part of the question.
That was the major part of the question.
Okay.
It wasn't the effectiveness.
Apologies for that. I can basically.
No, just give me your recall, because that's all we can do about influenza seasons. They tend to blend over time.
We have had, I think the main message for this season is that we had an emergence of a new subclade that hadn't been there previously that does have some genetic differences. There is at least some data from at least one study in Thailand that seems to indicate that the severity was not, as far as being required to be hospitalized, was not different from the other circulating strains for the season. The VE estimates we have are similar to those for other countries that have made VE estimates. differences from last season in terms of VE, basically comparing vision estimates and US flu VE estimates, which are the networks that we have comparable data for adults, for both overall adults and 65 plus. In most cases, if you're comparing apples and oranges, similar age group, similar setting, the estimates were between 10 and 14% higher, with the exception of those 65 and older, for whom this year's VE was actually better. So that is what I've assembled for the first question, and then I also have some information for Drs. Gans and Munoz.
Could you please tell us, because those are important questions.
Yes. So with regard to the questions Dr. Gans and Munoz asks, seem to revolve around relationships between coverage and other indices of severity and how they relate to severity in children. Just considering how these things relate overall. For coverage, the most recent data that we have that are complete actually finished with the end of last season, 24-25. And I can confirm, as I mentioned earlier, that if you look at the seasons going from 21-22, at which time coverage kind of peaked, down to 24-25, we have stepwise decreases in pretty much all of the age groups. 65 and up went from about 74% to 4 years later, 63.8%. Data in children similar, 21-22 for the entire pediatric age group, 51.4 to 43.8. So we know there have been decreases in coverage. As far as how these relate to severity, CDC assesses severity using surveillance data, using a number of indices, ILI, hospitalizations, flu-associated mortality, basically as a way to compare one season with another season. And when we consider things like overall burden on a population level, there are probably other things that go into that interplay in a very complex manner. VE, coverage, which was mentioned, as well as things such as perhaps age cohort effect, previous immunity, and the kind of kinetics of how flu activity is over the course of the season. This season, we have somewhat lower VE than last season. This season is overall moderate, severe in kids. Last season, 24-25, we had a season that was rated from our surveillance data as severe across the board for all age groups. We had considerably higher cumulative hospitalization rates for all of the age groups. And we also had during that season sort of a longer period of intense flu activity, with basically more weeks of active flu activity, more weeks of many percentages of specimens being positive, which allows for more accumulation of illness. So I guess I would say it's sort of difficult to kind of predict or assess the severity of one season for another, and the reasons why based on just one parameter, whether it's VE or something else. But that basically is the sum of my answer to that question. I hope that helps.
Thank you. Dr. Munoz.
Thank you, Dr. Manto, and thank you, Dr. Grosko, for this additional information. I have two questions for Moderna, and one relates to one of the advantages or the rationale for the mRNA vaccine is the time of production, the shortened times of production. And so one question relates to, if you could state what that time could be. And for example, is this a vaccine that if available and approved, that if approval will be available this season? And it's 2026-27.
Yeah, so I'll bring up our our COVID timeline as an example, and then I'll give it to Dr. Alan Embry, who's our program leader.
Good afternoon. I'm Alan Embry. I'm the program lead for the influenza program here at Moderna. Thanks so much for the question. So yes, as you said, we've talked a lot about how the mRNA platform allows for the more rapid design and incorporation of antigens without the reliance on viral reassortment and propagation in eggs or cells. And this allows us for the ability to rapidly transition from strain selection to manufacture and delivering to the public. For our seasonal COVID updates, as you can see on this slide, we've demonstrated over multiple seasons that variants, typically selected in the late May timeframe, can be authorized and available in a timeline of 2 to 3 months. And additionally, the completely synthetic mRNA platform allows us the ability to provide highly scalable production, so capable of meeting large population demands within these compressed timelines. So we're currently working with the FDA to establish, kind of the final regulatory package for our annual strain updates, and we'll know a little bit more once that's finalized. But we think together that this greater manufacturing speed and flexibility really provides the potential to significantly advance prevention against seasonal influenza and, if approved, for potential preparedness against pandemic vaccines such as H5N1. Our goal is to be able to deliver the vaccine this season, but that's for further negotiation with the FDA.
Thank you so much. May I ask another question? Just real quick.
Yes, go ahead.
Thank you. And so it relates to the phase 4 study, and it's very ambitious, very high population and 2 seasons. So regarding the specific goal, it's really to confirm. It's confirmatory for the efficacy. I understand that, but it's also this high-risk population. And so I was wondering if you are also considering, or it would be possible to include additional safety assessments and additional information regarding concomitant vaccination for these population, and particularly also the other effects that you can prevent from influenza-related diseases like myocardial infarction, strokes, and other things. Is that also something that you might be able to assess in terms of efficacy or effectiveness?
Yeah, indeed. And so first, I'll take this backwards then. So in terms of other endpoints, I think I think the methodology for these large trials is getting better and better and more streamlined. And I think this is enabling us to capture these other endpoints, which we know are really important from COVID and influenza and other respiratory vaccines. And so, these will be exploratory. I think the study will be powered on medically attended influenza and then the more severe directly influenza-related outcomes, but we can certainly look for these other things as well. Because this is so large and really happening in the real world, we will certainly allow for concomitant vaccination, and then we can capture that and then make an assessment. But we do believe that the concomitant vaccination will be, we have confidence in the concomitant vaccination because of the Spikevax flu vaccine data. And also we have a combination vaccine program where we've looked at these same mRNAs, the 1010 and the mNEX spike mRNA, and we didn't see any evidence of interference. And so it's out of the scope for today, but we do feel confident in those data. In terms of the immunocompromised recruitment, the recruitment will be broad. And given the sheer number, we expect to have immunocompromised populations here as well. And we will enable the sub-analyses.
Thank you so much.
Thank you. Dr. Gans, final question.
My final question. Thank you. I did have a question, I think along the lines of Dr. Muniz about what other populations. So we'll just bring up pregnant women as something down the line. I'm just going to stick that out there. It's not a question. My question is, did you have a chart? I know you showed overall the efficacy over the influenza season of the breakthrough disease, but did you have time to infection? Are there clustering? So for instance, in the messenger RNA vaccine, where the cases accumulated throughout, so it didn't really show that there was a waning of immunity towards the end of the season versus the comparator that may be clustered more closely with a remote time to vaccination. So I wondered if you're collecting that data and how the cases accumulate so we can get some understanding of persistence of immunity from that data.
Yeah, so I will take these questions in reverse order again. So, in terms of the case accumulation, so this is our overall accumulation curve, and we also looked at this very carefully, kind of month by month, and there was not a clustering. If you're asking if mRNA, if the slower case accrual in mRNA kind of was lost at some point. We did not see that on a month by month analysis. The accumulation really followed the epidemiology. The peak influenza season timing was when we accrued most of our cases. So like January, February, March was when we accrued most of our cases. In terms of the pregnant women data, we have, so this is 50 plus, but we have done our study in 18 to 49 for immunogenicity. I don't believe we had any pregnancies. We did have some pregnancies in those studies. Apologies. We did have some pregnancies in those studies, but it was just 17, and there was no adverse signal there as well. But certainly appreciate the comment that you made before about vaccination of pregnant women to prevent disease in young children.
Thank you. Dr. Berger.
Hi. A question for Moderna here. I just wanted to ask, I know you had a very low accrual rate for those individuals with influenza B. The confidence intervals were quite wide and across zero. I know the point estimates were in line with the others, but it did leave me to question, are you intending to collect additional efficacy data for mRNA 1010 against influenza B? And I note this just to say the last season we saw an increasing rate of B coming up at the end there. So it is something to potentially be concerned about going forward.
Yes, indeed. And so I'll just bring up our by strain efficacy and because of the high severity season for influenza A, we were actually able to get statistical significance for H1N1 and H3N2, which doesn't happen in all the seasons that flu studies are conducted. But for B, there were 60 cases, and the point estimate is very promising and consistent with the immunogenicity, but indeed it is wide. So we are going to do type specific efficacy in the PM and the post-marketing requirement study. So including influenza B, I think how diagnostics has moved forward with the actual strain confirmation and typing will allow us to do that. And so we will have those data.
Thank you.
Thank you. And thank you to Moderna for very clear presentation. So we next will move on to the committee discussion and voting. We will be voting separately on the two questions, the BLA and the accelerated approval. But I think in our discussion, we can look at both of the topics collectively, since there are data from the first study, which will give us some help in the accelerated approval. So please raise your hands. I do not see any hands raised. Have they said it all?
Did you want us to just deliberate amongst each other, or?
I think we should deliberate among each other about that. And why don't you go ahead?
Okay. So I think this there's a particular issue when a study is planned for 2 years and then, you know, for statistical reason, it's cut short on year one. Sometimes that's needed in an emergency to grant approvals to save lives in the middle of a pandemic. Sometimes it may end up also licensing a product while, you know, the study keeps continuing and gathering blinded data in the interim. So given the preceding season where the superior immunogenicity of the A strains was not observed, I think there was a big missed opportunity here by cutting the study short. Although, granted, the statistical analysis is sound on the first season in terms of what was observed. So that sort of makes, I'm not going to use the word doubt, but it sort of gives a pause, you know, comparing the preceding season and the 24-25 data on the flu A. So there is that point, and I don't know what the impact will be on of the increased reacto, grade 3 reacto specifically, in this vaccine, because we are talking about working age individuals, 50 to 64, are part of the group in whom this vaccine will be licensed. And 2 to 4% of people not going to work for a day or two is not trivial when we're talking about millions of individuals. And the unintended consequences on the vaccine programs, which I know as FDA, we don't deliberate much about here, as opposed to, say, ACIP. But it'd be important to be prepared with contingencies around this particular question. The planned study, the 400,000 individual study, will be important to answer some of the issues pertaining to efficacy and immunogenicity, but it's in older than 65. The 50 to 64 remains unknown. And so, yeah, these are the thoughts or the discussion points I wanted to bring up.
But of interest, there were significant differences in especially systemic side effects in even the immunogenicity study between the groups. So I think we're going to get a lot more information about it, even in the 65 plus population, because to me, this is the big question.
And it is a pattern across, you know, antigens, not just, and it's across manufacturer. Exactly. Because these are almost the exact same reacto data in the Pfizer product that was published in the New England.
Exactly.
8 months ago or so, yeah.
Dr. Durbin.
Yeah, just sort of in my summary. One, I do want to just highlight that the study was not ended early. The endpoint of the study was accumulation of cases, and they got to those cases quicker than they did. So I would just be careful about characterizing it as stopping the study early, because they didn't really stop it early. They met their endpoint sooner than expected. And it would have been nice to see, to see efficacy over 2 seasons. That would just be 2 different vaccinations, and it may or may not give us a lot more data. I think it's the data that they presented, and just my takeaway from the data that they presented for both 50 to 64 and greater than 64. I'll start with the 50 to 64. I thought the comparator data were very encouraging. I think the vaccine looks excellent in terms of its effectiveness, comparative effectiveness to standard dose vaccine. I think the adverse events that we're seeing are very comparable across other mRNA, Moderna specifically mRNA vaccines. I do think that that is something that the company is going to have to address. Certainly, we know that the best pathway forward is transparency, and I think we see this with other vaccines such as Shingrix, which are highly reactogenic. But it is something the company, I think, is going to have to address head on. And I liked Dr. Ganz's approach too, is, when we talk about reactogenicity and what causes that, bringing that into the discussion in terms of possible relatedness to effectiveness. In terms of the 65 and older, I thought the immunogenicity data were very compelling. I thought that it gave me confidence that this vaccine would be effective in 65 and older, would be at least as effective as current enhanced vaccines. We do need, of course, the phase 4 post-licensure study to demonstrate that. But I think in terms of safety, immunogenicity, and effectiveness, the vaccine looks very promising. And if it can meet its potential to be a reactive or to be able to change strains quickly, depending on epidemiology, I think that can be a real, very important additional benefit to this vaccine. Thank you.
Thank you, Dr. Durbin, and thank you for moving us to the next, partially into the next phase, which is explaining how we're going to be voting, because that's the whole reason for having this discussion, to try to get an interactive feeling about how we're going to act. Dr. Ganz.
You always sound surprised to see me, Arnold.
Welcome back.
Anyway, thank you. It's good to be here. Anyway, I just wanted to iterate some of the comments by Dr. Durbin, because I do think that the data that was presented and her synopsis is very good. I agree it was not stopped early, and I think that it's important that they met their time points. I do think we will learn more.
And the problem is with flu, you may go 2 years and still see a paucity of cases of one type or subtype.
Yeah, so I see Hannah's raised her hand again, so I'm very anxious for her to educate us. But anyway, if there's something additional that she's really worried about, my biggest excitement about this particular vaccine is that it actually induces better T-cell responses. I think we have been in all of our VRBPAC meetings and sort of struggling with the current influenza vaccines because they don't maintain great immunity. And this, to me, actually is one of the better components of how we're going to elicit better immunity across our population. And maybe even moving into, do we need actually annual vaccines? So this would be a really great advancement for everybody. So that to me actually is very compelling. And I do agree that I feel like the safety data that was presented actually, while it is true that nobody wants to be incapacitated, I do think that if it amounts to a stronger immune response, therefore better protection, if they have the luxury of being able to do it on a weekend or whatever, I don't know. So I do think if we message that right, that to me, what is also very promising to me is that in any of these newly licensed, we get very good follow-up surveillance. So as we've also discussed on this, and I think the sort of community really needs to understand, the public, that our surveillance systems are very good now for detecting signals and then studying causality. So unlike the days of just relying on VAERS, which is an important signal to notify us, we then can go back and look at these larger populations. So that to me also feels that with the current very safe data that we already saw, yes, there's discomfort, but I feel like we will get more and more data. And I think the other message we need to message out to our public is that the messenger RNA base of this has been used in billions of different vaccines now, since it's the same particle derivation that we have for the COVID vaccines. All important messaging for the public, but I really feel very strongly if we can induce memory responses, that's a huge advantage to this vaccine.
Thank you. Dr. El-Sali, did you have any, were you adding to what we just heard?
Yes, pertaining to the issue of terminating the study early. Yes, the study is event-driven and it did meet the event numbers that is pre-planned. So the word I used is terminated early is probably not accurate, so I agree. But the study was planned for 2 years. And given the unpredictability of flu and given what was observed in the year preceding the current study, it would have been wiser to continue the study. That's all I wanted to say.
Okay, thank you. Dr. Omer.
So 2 things. First of all, sorry to be a party pooper, but I would say that we should be careful about messaging reactogenicity as a de facto surrogate for efficacy, because the evidence across multiple vaccine is not, at the level where we could say that, especially coming out of regulatory deliberations. What we could say at best is that generally if vaccines are more reactogenic, they tend to be associated, and again, the operative word is associated, with more innate immune activation. But then there are 5 steps from there to clinical protection, and even that is a correlative relationship. So we should be careful about that. The second thing is, this is not to say that the data looks strong in terms of the overall safety and the risk-benefit calculus, et cetera. I would, as we move toward a vote, I would re-emphasize, given the framework that was reiterated in the morning by our FDA colleagues, under which we are to deliberate, that the imbalance in all-cause mortality does not warrant at this point any sort of further interpretation of these data. But it does, from my perspective, from looking at the uncertainty part and especially accelerated pathway, it does warrant a very intentional assessment of all-cause mortality in the planned post-marketing study as an a priori outcome, so that hopefully it will put sort of this any concerns to rest, if any, but also that's the right thing to do. And the more appropriate thing to do. And so we are talking about a pretty large study, which is 400,000. But if it's 2 years, so we are looking at 800,000, nearly 1 million person years, or 800,000 person years. So that we should be able to answer those questions.
Thank you. Dr. Berger.
Thanks, Dr. Monto. I think most of the things I'm going to say have already been said, so I'm going to try and do it succinctly. There are a number of limitations here. This was a study that was conducted over 1 season. There's limited efficacy data against B strain. The efficacy data in 65 plus used a non-preferred comparator, and there's no data on immunocompromised and limited data on very frail older adults. And clearly there's a need for follow-up on durability and rare adverse events. The immunogenicity data, however, is very suggestive of efficacy or potential efficacy clinically, and it's great to see the higher efficacy compared to existing vaccines. And the safety data is relatively reassuring overall. Overall, I think the evidence largely supports safety and effectiveness here. I think it does leave some questions for FDA to really consider how they would implement a new or different timeline that would not deleteriously affect access to other non-mRNA-based vaccines. And for Moderna, it's going to be essential that they ensure the post-marketing study really addresses a lot of the remaining uncertainties that I just laid out and that others have noted here. And I'll just, again, say particularly for those that are elderly and frail adults and immunocompromised populations. Thank you.
Thank you. And I appreciate moving into explanation of why you're going to be voting the way you will. That way we can avoid having an explanation of votes after the vote. Dr. Patel.
Thank you. I just wanted to add 2 comments related with a public health lens to the conversation and agree with what the other members have said. So one is that from a pandemic preparedness space, this is an important advancement to continue to build and grow. And so appreciate the Moderna and others actually continuing to think about mRNA vaccines in that context. The other thing, and this was mentioned, Dr. Groskopf mentioned this, that with decreasing vaccination coverage, I worry a little bit that we're going to meet the endpoints in the phase 4 trials. And so just for Moderna to think through, if enrollment is low, what are the other options that they have and sort of contingency plans for that? So thank you.
Okay. Just don't say how you're going to vote. Just explain why you support or do not support in this discussion. Okay, Dr. Berger.
Sorry, Dr. Mann, I was just have one more thought I wanted to raise, which is just, more of a general comment, but, we have decades of data on the safety of existing influenza vaccines. We don't have the same thing for mRNA vaccines. And I just want to throw it out there that it's going to be very important that we create or collect that information and start generating the evidence base to demonstrate that these have long-term or demonstrate that long-term these are safe to use on a yearly basis. I'm not saying there's anything wrong with them. I'm just saying we need to have a collective effort to really have that data generated and assessed.
Thank you. Any other further comment? Dr. Perlman.
Something I've been mulling while we've been talking, and you can tell me this is completely off what we should be thinking about, but do we know if this is going to add to healthcare costs? Is this vaccine going to be priced similarly to others?
That's a question. I don't know if the Moderna people are still around. If they are, please respond.
Sure. So unfortunately, I can't provide any details on that. There's no, we don't have any pricing data yet.
Okay, thank you. Dr. Durbin?
Dr. Mato, I would think that would be more of an ACIP question as opposed to an FDA question, but maybe FDA can answer that for Dr. Perlman.
If you can, please.
Yeah, just very succinctly, I think Dr. Durbin answered the question. It really is not an FDA question.
Hello, this is Mary with Tax Consultants of America. Today is Thursday, June 18th. I'm calling regarding a time.
Sorry, I was having an iPad issue. Seeing no hands, I'm rather amazed. Then I think what we should be doing is getting, if you are all ready, Cecily, for voting questions, we might as well just move on to the voting questions.
Certainly. Thank you, Dr. Monto and the committee and everyone for your deliberations. The questions before the committee today are both voting questions. Voting members will use the Zoom platform to submit their votes for this question. Votes for both questions will be cast together during the voting session. If you are not a voting member, you will be moved to a breakout room while we conduct the vote. After Dr. Monto reads the voting question into the record and all clarifying questions are complete, we will announce that voting will begin. A voting window will appear where you can submit your vote. There will be no discussion during the voting session. You should select the button in the window that corresponds to your vote, yes, no, or abstain. Please note that once you click the submit button, you will not be able to change your vote. Once all voting members have selected their vote, I will announce that the vote is closed. Please note there will be a momentary pause as we tally the vote results and return non-voting members into the meeting room. Next, the voting results will be displayed on the screen, and I will read the vote results from the screen into the record. Thereafter, Dr. Monto will go down the list, and each voting member will state his or her name and how they voted on the two votes into the record. Voting members should address their rationale for their vote. Dr. Monto, would you please read the first question into the record and followed by the second question? Thank you.
So the ballot will contain both voting questions?
Yes.
Okay. For the record, do the benefits of Influenza outweigh its risk for the prevention of influenza disease in adults 50 through 64 years of age? And number two, do the benefits of Influenza outweigh its risks for the prevention of influenza disease in adults 65 years of age and older? And you can vote yes or no or abstain.
Thank you. We will now begin voting. Thank you, everyone, for your patience as we tally the voting results. We'll wait to have the voting results displayed. So we had, for both questions, we had 9 yeses, 0 noes, 0 abstained. And for question 2, we had 9 yeses, 0 noes, and 0 abstained. Dr. Monto, I'll hand over to you, and you can go down the list and have each person detail how they voted for both questions and discuss as necessary.
Okay, and yes, that's a good idea. If you would like to explain your vote as you go down the list, there's no requirement. Please do so. Dr. Berger.
I voted yes in both for both questions. Nothing to add from what I said previously.
Dr. Patel.
Similarly, I also voted both. I think the relative VE in the 50 to 64-year-olds and the immunogenicity data in the 50 plus are promising. I acknowledge that there's increased reactogenicity in the mRNA 1010 groups, but there wasn't appear any major safety signals. So that was also reassuring. I did struggle with the lack of a relative VE that data for the 65 plus, but if we follow the framework that Dr. Caslow had put forward, I think that the benefits outweigh the risk for that accelerated approval for that particular age group. And just want to acknowledge comments about the limitations and additional data that are needed. I think Dr. Berger had nicely summarized that in the session before the vote.
Dr. El-Sali.
Yes, I did vote yes on both counts. The presented data do indicate efficacy against flu with no safety signals. Hence, that's why I voted. The limitations, I already previously indicated those. The increased reactogenicity needs to be carefully addressed and messaged to the public, especially as it pertains to the issue of vaccine acceptability in general.
And I see I'm next. I voted yes to both of the questions, and I want to commend both the sponsor and FDA in working out the accelerated approval, which took care of my concern about the comparator in the pivotal study. So, Dr. Munoz.
Yes, I voted yes to both questions because I do believe that the data presented support that the benefits of the vaccine in both age groups outweigh the risks that as have been discussed. I think that the studies that were presented today were very well conducted. They have very clear results that are very robust in terms of demonstrated that additional efficacy above what we know existing standard dose in the case of 65 and older, although a smaller study, higher dose vaccines can provide. So I think that in addition to the stated benefits for individuals who are going to have this potential higher efficacy support, we have the rapid development of the vaccines in regular seasonal flu activity and changes of strains. But this also puts us in a position, having this technology available, to be better prepared for emerging strains or pandemic strains in the future. So I think this is also important to keep in mind. And lastly, I agree with the very nice work coordination that was done between the applicant and the FDA in terms of providing the option for these very well planned and large phase 4 study that will continue to address important questions. And I would definitely urge the manufacturer to address not just the effectiveness data, but also looking at additional potential benefits of this vaccine, and especially the older populations. So thank you.
Dr. Perlman.
Yeah, I don't have much to add. I think that the data were very convincing, and I also think that the FDA, CDC, and sponsors did a very good job in presenting the data. I will be very interested in the subsequent study, both for older people and then also as this vaccine is being used in younger populations. I'm really curious as to whether there's going to be any signal of myocarditis in 17 to 30-year-old males, because we really don't have any information about that, and we don't really know why the mRNA vaccines seem to be associated with that. But that's why I voted yes, because putting it all together, the benefits outweigh the problems, the risks, in my opinion.
Dr. Gans.
Thank you. I voted yes to both of the questions before us. I think that this particular platform adds exciting ways that we can actually move our vaccines to the future. And I am also additionally sensitive to all of this safety that will go into the post-marketing. The signals that we're seeing now are not putting people at risk, and the benefits are actually large, not only for this season, but for really what it can do for our vaccine platform. So I think we continue to monitor, we continue to do further studies, and I think we'll learn much more. And this one has the potential to really move us in modern direction. So I voted yes.
Dr. Omer.
Yeah, I also voted yes on both questions. And the context in which I voted and the way I voted is already part of the record, during the, based on the comments I shared in previous sessions, so I have nothing further to add. And thank you for capably coordinating this meeting, and this is an opportunity for me to thank the staff and the sponsor for a very well-conducted process.
Dr. Durbin.
I also just want to thank the FDA and Moderna for really very, very nice presentations. I voted yes for both questions. The only couple of thoughts that I had after that I would add is that I thought, the data, the effectiveness data using the standard comparator, when we look even at 65 and older, was similar effectiveness to what the enhanced dose vaccines were able to obtain licensure. So that gave me some confidence in addition to the immunogenicity data that were presented in the 303C study. The other thought that I had is having a single vaccine for all adults 50 and older, again, may have an impact on vaccine availability and ease of manufacture, which can be helpful in future flu seasons as well. And that's all I have to add. Thank you.
Thank you all for your contributions and your votes, which clearly speak for themselves. I believe that closes my part of the meeting, and I'd like to turn this back to Dr. Kaslow and to Dr. Reed, who will close the meeting.
Great. Thank you, Dr. Monto. So let me start with just a few brief reflections before ending with some thank yous. Today's VRBPAC convened for the 193rd time, convened to consider and discuss in a transparent, open public session, a large amount of evidence that informs the potential benefits and risks of Influenza. The office noted several points made, including about correlates of protection, about the current process for seasonal influenza formula updates in the context of mRNA manufacturing technologies, about communicating the benefits and risks, which is actually the work ahead in this BLA review on the proposed prescribing information, about the additional outcomes beyond acute symptoms and signs of influenza disease and evidence in special populations, about single season data, and about the added value of the proposed phase 4 confirmatory study. So thank you again for yet another robust evidence-driven discussion to drive improving public health against this serious and life-threatening pathogen and to enhancing public trust in vaccines. So I'll end by first thanking the open public hearing speakers, the private citizens, and the organizations that posted comments to the docket. Let me also thank all of the presenters for their outstanding interventions today. You drove a robust public discussion, noting both the strengths and the gaps in the evidence. The setting for those presentations would not have occurred without a large team here at FDA to host another flawless VRBPAC. So thank you to the FDA Advisory Committee staff, our AV staff, and my colleagues in CBER and OVRR. And finally, a sincere thanks to today's VRBPAC members for your courage in the name of public service. It is vital for enhancing public trust. And thank you, Dr. Monto, for your service as today's chair. So with that, I will turn the meeting back to our DFO to adjourn the meeting.
Thank you, Dr. Kaslow. I'll adjourn the meeting at 2:53 p.m.
Thank you, everyone.
Thanks.
Thank you.
Thank you.
Thank you.
Thank you all.
Thank you.